In the quiet space of imagination, it helps to picture Earth not as the familiar, living world we know today, but as a slowly cooling sphere drifting through the darkness of early space, wrapped in shifting clouds of vapor and illuminated only by the softened glow of a younger, gentler sun. In those earliest ages, no life stirred upon the surface, and nothing yet breathed or reached or swam. Instead, the world was defined by stillness and time. Everything moved slowly, from the settling of molten surfaces to the formation of the first gentle rains. Volcanoes released warm plumes into the sky, while oceans forming below them simmered with minerals, drifting quietly from one place to another. It was a world without sound, except for the steady movement of water and the distant rumble of the Earth's cooling crust, creating landscapes that waited patiently for something more. The early crust had begun to harden, forming continents not yet shaped into familiar outlines. As the molten rock below found its calm, the first stable surfaces held firm, cooling further with each passing millennium. Rain, which once evaporated instantly under the planet's intense heat, began to linger long enough to collect. At first, it formed shallow pools, and later it merged into broader basins that spread into the earliest oceans. These waters were warm and mineral-rich, filled with dissolved elements and stirred by tides that were far stronger than those we know now. Under the hazy sky, the light filtering down was soft and diffuse, muted by volcanic gases and tiny particles suspended in the air. The world glowed faintly beneath this veil, illuminated in tones more subdued than daylight, creating an atmosphere that felt hushed and waiting. Even in this quiet environment, the building blocks of life were already in motion. Earth's oceans acted as natural vessels for carbon, hydrogen, nitrogen, and oxygen, the elemental threads that would eventually weave into more elaborate structures. These atoms drifted freely through the water, colliding gently, forming simple molecules, separating again, and slowly reorganizing. Over thousands upon thousands of years, these interactions accumulated. Some molecules grew long enough to fold into interesting shapes. Others remained small but stable, waiting for the right combination of heat, light, or minerals to change them. Energy from sunlight warmed the surface of the oceans, while flashes of lightning threaded through the sky, briefly lighting entire regions of water with bursts of power capable of shifting chemical bonds in an instant. Meanwhile, geothermal vents beneath the surface released warm, mineral-rich fluids that mixed with the surrounding seawater. Each of these energy sources encouraged reactions that would otherwise not occur, creating a quiet chemistry lesson unfolding on a planetary scale. During these long stretches of time, the basic principles that life would one day rely upon were coming into focus, even though no organism yet existed to make use of them. Life depends on the ability to maintain internal order, to use energy, to respond gently to changing conditions. These central features, organization, energy use, homeostasis, reproduction, and adaptation, were not yet traits of living organisms, but they were emerging as potential pathways, shaped by the behavior of molecules that persisted longer than others or reacted in more stable ways. By understanding these foundational features now, softly and without urgency, we can begin to appreciate the extraordinary transition that was about to take place on the early Earth. Minerals continued flowing into the oceans as rain washed over the forming crust. Iron, sulfur, phosphorus, and other essential elements drifted into the water, becoming part of the underwater chemistry. Hydrothermal vents played an especially important role, releasing warm, dark fluids filled with metal ions and gases that blended with the cooler water above them. These vents created steep temperature gradients and chemical contrasts, conditions known to encourage the formation of complex molecules. When warm water rising from the vents met the surrounding ocean, it cooled and deposited mineral structures that resembled chimneys or towers. Inside and around these mineral formations, the chemistry of early Earth took on a new dimension. Some scientists believe that these environments may have offered the perfect balance of stability and change, enough protection for fragile molecules to form, and enough energy to encourage them to evolve into more complex shapes. Earth itself was perfectly positioned to nurture these gentle beginnings. Its distance from the sun allowed water to remain liquid across large parts of the surface, a rarity in our solar system. The planet was large enough to hold a substantial atmosphere, which filtered out some harmful radiation while still allowing the exchange of heat. Water, acting as both a cushion and a medium, protected delicate molecules from immediate destruction and encouraged their slow refinement. The rotation of the planet produced cycles of light and darkness, helping regulate temperatures and keeping the oceans in motion. These natural rhythms, long before life began, would later echo in the daily cycles that shape the behavior of organisms, from plants opening in the morning light to animals resting through the night. To imagine these early ages is to imagine deep patience. Nothing rushed. Chemical reactions did not know the concept of time. A single change in a molecule might take place in a moment, but the accumulation of meaningful patterns required eons. The planet worked quietly, steadily, without haste. With each passing millennium, new molecular arrangements became possible. The oceans grew richer, not with life, but with possibility. Each molecule that proved slightly more stable or slightly more adaptable contributed to an expanding library of forms, each one a potential step toward what would eventually become a living cell. As these slow processes continued, the environment of Earth moved ever closer to a turning point. Organic molecules, those containing carbon and capable of forming long chains, became increasingly common in the water. They bonded with one another in ways that made them harder to break apart. Some began to form structures that could fold or maintain their shapes across changing temperatures. Others grew into chains capable of binding smaller molecules to themselves. Earth had prepared a set of ingredients, and through gentle, continuous reactions, the oceans had begun to stir those ingredients into arrangements that carried the promise of something new. Eventually, after spans of time so long they are almost beyond human understanding, conditions reached a delicate balance. The oceans held warmth, minerals, and energy. Molecules had grown complex enough to perform limited functions. The rhythm of Earth's cycles continued steadily, and somewhere within that vast and shimmering expanse, the first hints of what would one day be life began to coalesce. The story had not yet fully begun, but its first chapter had taken shape in the soft quiet of the young Earth. Before anything breathed or moved with intent, the foundation for everything that would follow had been laid, patiently and peacefully, awaiting the dawn of biological complexity. 2. The foundations of life order and stability. When we imagine the earliest beginnings of life, it is soothing to pause for a moment and reflect on the quiet principles that would one day guide every living organism on Earth. Before there were cells, before there were plants or animals, or even the simplest microbes, there were the conceptual foundations that life would eventually adopt: organization, responsiveness, energy use, growth, reproduction, adaptation, and the ability to maintain internal balance. These features did not appear instantaneously, nor did they emerge in perfect form. Instead, they arose gradually, shaped by chemistry, long before biology took hold. In the soft unfolding of the planet's early history, these traits began to form the invisible scaffolding upon which the first living systems would later be built. At this stage of Earth's story, it is comforting to think of these principles as quiet ideas taking shape in a world not yet populated. The first of these foundational characteristics is organization. Even the simplest forms of life today display an extraordinary level of structure, from the arrangement of molecules within a cell to the organization of tissues and organs in more complex beings. Yet the earliest organization on Earth was not biological but chemical. Molecules assembled in patterns, some forming chains, some folding into shapes that allowed them to interact with their surroundings in consistent ways. This early ordering created pockets of stability, places where molecules could persist long enough to undergo further reactions. These small moments of structure were significant, for they laid the groundwork for systems that would one day maintain boundaries, regulate internal conditions, and conduct organized activities. Closely tied to organization is sensitivity, the ability to respond to changes in the environment. Even long before true responsiveness emerged, early molecules interacted with their surroundings in subtle ways. Temperature shifts altered their stability. Light from the sun provided energy to excite electrons, and mineral-rich water influenced how they bound to one another. These early interactions foreshadowed the more refined sensitivity seen in living organisms today. Plants bend gently toward the sun, pupils adjust softly to changes in light, even single-celled organisms swim toward nutrients or away from danger. All of these behaviors trace back to that early, primitive responsiveness, when the environment shaped molecules, and molecules, in turn, shaped the environment. Growth, development, and reproduction, another cornerstone of life, also have deep roots in early chemistry. Modern cells grow by producing new molecules and replicating their internal structures. But the earliest forms of growth were far simpler. Chemical chains lengthened as new elements attached. Molecules that were stable enough to persist through environmental changes accumulated gradually in the water. Over time, these longer structures became more capable of performing the kinds of functions associated with primitive metabolism. In this sense, growth did not require a living organism to begin. It was a gentle, automatic process, guided by the availability of resources and the stability of particular molecular arrangements. Development arrived later, as molecules began forming systems that could change their shape or behavior in response to conditions. Reproduction, the ability to create copies, was still far in the future, but its earliest hints were beginning to appear as certain molecules gained the ability to replicate portions of themselves, marking the transition from chemistry to something more lifelike. Energy use, another defining feature of living beings, was also present in the early Earth in a quiet and unintentional form. Today, all living things rely on energy. Animals obtain it through food, plants through sunlight, and even the tiniest cells through chemical reactions carefully orchestrated within their membranes. But in Earth's early oceans, energy flowed freely and continuously, powering reactions without the guidance of a living structure. Sunlight warmed the surface waters, breaking some molecular bonds while strengthening others. Lightning provided short bursts of high energy, capable of rearranging atoms in dramatic ways. Thermal vents on the ocean floor emitted steady streams of heat and minerals, creating gradients that encouraged molecules to form and interact. These energy sources charged the environment with the potential for change, creating conditions in which the fundamental processes of life, metabolism, transformation, and motion could eventually take shape. One of the most essential features of life, homeostasis. Homeostasis refers to the ability of an organism to maintain a stable internal environment despite external fluctuations. Humans regulate body temperature, plants control water loss, and single-celled organisms adjust their internal chemistry to survive. In the early oceans, stability came not from organisms but from the environment itself. Water acted as a buffer, providing a consistent medium that protected delicate molecules from rapid temperature changes. Minerals dissolved in the oceans created an environment with reliable chemical conditions. Without this natural stabilization, fragile molecules might never have persisted long enough to become part of something living. Though true homeostasis would emerge only with the first cells, the ocean's gentle consistency offered an early form of the protection that life would later learn to create for itself. Among the most compelling qualities of life is evolution, the gradual change of populations over time. This, too, had its earliest beginnings in the slow, continuous transformations of prebiotic chemistry. Molecules that were more stable tended to persist, while unstable arrangements fell apart. Some molecules reacted more readily with others, forming structures that were better suited to the conditions around them. Over time, these simple patterns of persistence and transformation laid the foundation for natural selection. The earliest forms of variation did not come from mutations or genetic recombination, as they do in modern organisms, but from randomness in chemical reactions. This randomness provided diversity, and diversity offered opportunities for certain molecules to outlast others. Through this gentle, unintentional filtering process, the stage was set for evolution to become a guiding force in the history of life. The principle of emergent properties also finds its roots in these primordial conditions. Today, emergent properties are seen in every level of biological organization. A single molecule cannot think, but a network of neurons can produce consciousness. A strand of DNA cannot survive alone, but within a cell, it becomes part of a dynamic and self-sustaining system. In the early oceans, emergent behaviors arose when molecules interacted in ways that produced new capabilities. A chain of amino acids, for example, might fold into a shape that allowed it to catalyze a reaction. A simple membrane might trap molecules together, allowing interactions that would not occur in open water. These outcomes were not the result of planning but of chance and chemical compatibility, yet they provided the earliest glimpses of the complex behaviors that would later define living organisms. As the principles of life began to take shape, the Earth itself continued to provide the environment needed for increasingly complex chemistry. The atmosphere thickened gradually, shielding the surface from harsh solar radiation. The crust cooled further, becoming more stable. Oceans deepened and expanded, mixing constantly through tides and currents. These quiet, steady changes created a world increasingly suitable for the transition from simple molecules to self-maintaining systems. Life had not yet emerged, but everything necessary for its arrival was falling gently into place. With time, the building blocks of life, molecules capable of forming membranes, storing information, and catalyzing reactions, would begin to appear. But before the story of life could truly begin, the foundations had to be set. These foundational principles, now universal across all living organisms, arose not through sudden transformation, but through slow, peaceful processes guided by the interplay of chemistry and environment. The Earth, still young and quiet, continued to nurture these possibilities, offering stability, warmth, and energy to the waters where life's first whispers would eventually be heard. 3. Ancient oceans as cradles of chemistry. To step gently into the third part of this story, it is helpful to imagine the early Earth not in geological chaos, but in a long, slow exhalation. The planet had finally cooled enough for its first oceans to settle into place, spreading like a soft blue veil across the rugged surface. These oceans were warm, rich with minerals, and constantly shifting. They were not yet home to life, but they were already becoming something more than water, something closer to a cradle, a vast vessel, where the chemistry of possibility unfolded in patient waves. In these early waters, the foundations of biology were being prepared quietly, without intention, and without the presence of anything that could yet be called alive. The oceans were simply there, holding atoms in solution, letting them drift and collide, allowing gentle experiments driven by energy from the sun, from lightning, and from the Earth's interior. These oceans were nothing like our modern seas. They were denser with dissolved materials, enriched by constant volcanic activity that poured minerals into the water. Iron, sulfur, and phosphorus swirled through the depths, forming gradients and pockets where different reactions could take place. The atmosphere above them, still thick with gases released from the planet's mantle, filtered the sunlight in subtle ways, letting only a softened glow reach the surface. This light warmed the shallow regions, while deeper waters remained cool and dark, creating a variety of environments all interconnected, all moving gently in response to the young planet's rotation. It was in this variability that chemical diversity began to flourish. Where light triggered reactions near the surface, darkness preserved delicate molecular structures below. The entire ocean became a dynamic canvas, allowing chemistry to take place in countless small, scattered regions. Lightning played a quiet but essential role as well. Great storms swept across the skies, sending flashes of energy into the water. Each bolt introduced enough power to rearrange atoms, breaking some bonds while forming others. These sudden bursts created opportunities for molecules that could not have formed otherwise. Meanwhile, hydrothermal vents on the ocean floor contributed their own unique energy. These vents released mineral-rich fluids heated by the planet's interior, rising into the cooler waters above and forming areas full of chemical contrast. In these gradients between hot and cold, acidic and alkaline, the earliest building blocks of life had the opportunity to form and persist. The vents created tube-like structures where minerals assembled naturally into porous walls, offering tiny compartments that could trap molecules together. These compartments may have acted as primitive reaction chambers, providing protection from the surrounding environment and concentrating materials that might otherwise have drifted apart. Within this dynamic environment, simple molecules gradually formed more complex structures. Carbon atoms linked into chains, forming the first organic compounds. Some of these chains bent or folded, responding to the surrounding water or to the minerals that touched their surfaces. Although none of these early molecules possessed life, some behaved in ways that hinted at what would come later. Certain structures became more stable in particular environments, lingering long enough to participate in future reactions. Others broke apart quickly, but left behind fragments that contributed to new combinations. Over millions of years, these processes produced a remarkable diversity of molecular shapes and sizes, each with its own potential. It was a slow accumulation, a gentle layering of complexity upon simplicity. The oceans supported these transformations not only through their chemistry but also through their natural stability. Water has a remarkable ability to protect fragile structures, cushioning them from sudden shifts in temperature or radiation. In the early Earth, this protection was crucial. Without the steady medium of the ocean, many delicate molecules would have broken apart as soon as they formed. Instead, they drifted safely through the water, meeting other molecules. Even if the earliest structures fell apart repeatedly, the ocean always offered a fresh environment for new combinations to arise. This continuous interplay helped shape the earliest steps toward life, allowing experimentation on a scale and over a duration impossible to replicate in any laboratory today. As these molecules drifted and interacted, certain patterns began to emerge. Some molecules naturally formed spheres or layers when placed in water, creating the first hints of compartmentalization. Others acted like rudimentary catalysts, encouraging reactions that would otherwise proceed too slowly to matter. These early catalytic molecules were not enzymes in the biological sense, but they performed a similar function, gently lowering the energy required for chemical reactions to occur. In doing so, they contributed to the growing complexity of the ocean's molecular landscape. Although these behaviors were primitive, they represented steps toward the core principles of life, toward the organization and controlled activity that living cells would later display. Over immense periods of time, with countless chemical interactions taking place simultaneously, the ocean developed an astonishing richness of organic compounds, each one a potential thread in the tapestry of life. The atmosphere above these oceans also played a subtle role. It contained gases such as methane, carbon dioxide, and nitrogen substances that interacted with sunlight and contributed to the chemistry of the oceans below. Some of these gases dissolved into the water, forming new chemical species that participated in molecular formation. Others reacted in the air, producing compounds that drifted gently downward and settled into the sea. The interplay between atmosphere and ocean was not rigid or constant, but it was steady enough to keep introducing new materials into the aquatic environment. In this way, the planet's surface became a continuous cycle of exchange and renewal. Nothing was static, but nothing moved at a pace that disrupted the delicate formations taking place in the water. As chemical diversity increased, so did the opportunity for new reactions. Molecules that once existed only briefly began to persist for longer periods. Structures that had been unstable a million years earlier started to hold together, forming the building blocks needed for still more complex chemistry. This abundance of stable molecules created the conditions necessary for the next major development in the story of life: the emergence of self-replication. Though replication itself had not yet begun in Part 3 of our journey, the oceans had become prepared for it. They had grown rich enough in organic compounds to support the creation of molecules capable of copying their own structures, an event that would mark the true turning point between chemistry and biology. Before that moment could arrive, however, the oceans needed to accumulate enough diversity, enough stability, and enough complexity. And so they continued to do exactly that, quietly, steadily, without urgency. In reflecting on this stage of Earth's history, it becomes clear that the oceans were not merely passive environments. They were gentle architects, shaping the earliest steps that would eventually lead to life. They provided the stability required for fragile structures to endure, the energy needed to drive reactions, and the diversity of conditions necessary for complexity to arise. Through countless cycles of formation and dissolution, the oceans transformed the raw materials of the planet into a rich mixture of organic molecules. In this cradle of warm, mineral-laden water, the planet prepared itself for the shift from chemistry to biology, from scattered molecules to self-sustaining systems. Everything that would unfold in later chapters began here, quietly and patiently, beneath the calm surface of the ancient sea. 4. The first self-replicating molecules. In the quiet, expansive oceans of the early Earth, long before the first cell took shape, an extraordinary transformation was slowly unfolding. The world had already produced a rich mixture of organic molecules, shaped by sunlight, geothermal energy, and the gentle movement of water. These molecules drifted through the seas like scattered pieces of a puzzle that had not yet found a clear pattern, but gradually, over spans of time far beyond anything humans can easily imagine, some of these molecules began to behave in ways that hinted at what life would one day become. They did not breathe or move or grow, but they possessed a subtle and powerful ability. They could make copies of themselves. This shift from molecules that simply existed to molecules capable of replication marked one of the most important transitions in the history of the planet. It was here, in these quiet seas, that chemistry began its slow and graceful transformation into biology. To understand why self-replication was such a profound development, it is useful to recall how life is defined today. Every living organism, no matter how simple or complex, relies on its ability to pass information forward, whether through cell division, flowering, or the countless reproductive strategies seen across the natural world. The continuity of life depends on the transmission of genetic material. But before the first organisms existed, there had to be a structure capable of storing information and copying it. That structure was not DNA, not at first, but something simpler, a molecule able to hold instructions and replicate in a basic way. Researchers believe that one of the earliest candidates for this role was RNA, a molecule still found in all living organisms today, playing central roles in protein synthesis and gene regulation. RNA is capable of folding into intricate shapes, storing information, and, under the right conditions, even catalyzing reactions. These qualities make it an appealing candidate for the earliest self-replicating systems on Earth. When we picture this early replication, it is calming to imagine it unfolding slowly and naturally, guided not by intelligence, but by chemistry. RNA molecules drifting in the ocean occasionally encountered nucleotide building blocks, simple chemical units capable of forming long chains. If a partially formed RNA strand provided a suitable template, these nucleotides aligned themselves in complementary sequences, joining together to create a new strand. This process did not occur with the precision or regularity of modern genetic replication, but it was enough. Over time, some strands reproduced more faithfully than others. Those that could fold into stable shapes or catalyze useful reactions persisted longer, increasing the likelihood that they would continue to replicate. Even without living cells, this gentle form of selection created a foundation upon which more complex systems could eventually emerge. The oceans provided the perfect environment for this early replication. Water protected fragile molecules from rapid degradation and brought reactants together in a continuous flow of gentle motion. Minerals present in the water may have acted as scaffolds, helping nucleotides assemble into longer chains. Clays, in particular, have been shown in laboratory studies to facilitate the formation of RNA-like molecules, offering surfaces where chemical building blocks can align more easily. Whether or not clays played a key role in Earth's earliest replication, the idea illustrates how the physical environment itself contributed to the rise of complexity. The planet was not passive; it offered resources, structure, and stability that supported the earliest experiments of molecular evolution. As replication grew more common, a quiet but profound process began to unfold. With each copying event came the possibility of variation. Some strands formed slightly different shapes, some folded more tightly or more loosely. Some attracted particular molecules more effectively, while others deteriorated more quickly. These differences were subtle, but they mattered. Variation created diversity, and diversity provided material for selection. In this way, replication and variation became intertwined, guiding the earliest steps into a realm that would later be shaped by natural selection. Even before the existence of true life, the seeds of evolution had begun to sprout. Yet replication alone was not enough to produce living systems. For replication to become a biological process, molecules needed ways to persist longer, to protect themselves from degradation, and eventually to interact with other structures in meaningful ways. This led to one of the most important transitions in early molecular evolution: the emergence of simple compartments. As certain molecules formed into natural spheres or layers, they created tiny boundaries, membrane-like structures capable of trapping, replicating molecules inside. These early compartments were not cells as we understand them, but they represented a crucial step toward the organization needed for life. By protecting replicating molecules and holding reactants in one place, they increased the likelihood that replication would continue. In a world without organisms, these microscopic compartments created environments that functioned like primitive, self-contained systems. Within these compartments, replication became more efficient. Molecules that catalyzed reactions could interact with those that stored information. Chains that had been scattered through the ocean now found themselves concentrated in pockets of stability. The simple membranes that enclosed these compartments may have formed from fatty acids, molecules that naturally arrange themselves into spheres when placed in water. These structures were fragile and easily disrupted, but they also formed readily and repaired themselves under the right conditions. Their behavior provided a preview of what future biological membranes would one day accomplish with far greater complexity and precision. The emergence of self-replication also set the stage for specialization. Some molecular structures were better at storing information, while others aided in catalysis. Over long periods, these roles became more distinct. Just as modern cells contain organelles that perform specific functions, these early systems began to show a division of labor on a molecular scale. Though still far from life, they had taken on characteristics that would eventually define biological systems: cooperation, stability, and functional diversity. These subtle developments helped the earliest molecular networks persist through changes in temperature, salinity, and other environmental conditions. In this way, the ocean's chemistry gradually shifted toward a direction that favored complexity. During this time, Earth continued its slow, steady cooling. The oceans deepened and became more chemically stable. These changes created a more consistent backdrop for the delicate molecular dance taking place beneath the waves. In such a world, replication could flourish. Molecules capable of making copies of themselves gained an advantage simply by persisting, and their presence shaped the chemical landscape around them. Bit by bit, self-replication introduced order into the ocean's otherwise random assortment of molecules. It did so without conscious intent, without urgency, and without awareness. It was a natural unfolding, a quiet transition from chemistry toward something more. Eventually, after millions of cycles of replication, variation, and persistence, the ocean contained a growing population of self-replicating molecules. Some survived better than others, forming more stable structures or catalyzing reactions more effectively. These small advantages accumulated over time, guiding molecular evolution toward increasing levels of organization. The earliest forms of heredity began to appear as certain sequences were copied more frequently. This was not heredity as modern organisms experience it, not yet, but it represented the first glimmer of continuity across generations. Molecules that had once been ephemeral now carried a kind of memory. They passed their structures forward, allowing successful patterns to endure. In this way, the ancient oceans became not only a cradle of chemistry but the birthplace of a new kind of order. They nurtured the transition from simple molecules to self-replicating systems, structures capable of persistence, variation, and inheritance. These qualities would one day define life. But here, in these early ages, they remained quiet and unrecognized. There were no cells, no tissues, no ecosystems. There were only molecules learning, in their own slow, unintentional way, how to copy themselves. And from this gentle beginning, the rest of life's vast and intricate story would eventually unfold. 5. The birth of primitive cells. In the quiet depths of Earth's early oceans, after millions of years of gentle chemical experimentation, the planet approached a moment of profound transition. The waters had grown rich with organic molecules, chains that folded into intricate shapes, clusters that clung together, and strands capable of copying themselves in a slow but steady rhythm. Yet for all their complexity, these molecules still drifted freely, unbounded and unprotected. They existed in an open environment where reactions occurred randomly, without direction or coordination. For life to take its next step, something new had to appear, a way to enclose these molecules, to create a boundary that would hold them together long enough for more elaborate systems to emerge. This step, subtle but essential, marked the birth of the earliest primitive cells, structures that were not alive in the modern sense, yet set the stage for everything life would one day become. To imagine this moment clearly, it helps to think of the ocean as a vast and peaceful laboratory where countless molecules mingled, separated, and formed new arrangements. Among these molecules were simple lipids, fatty substances that behaved differently from the surrounding water. Lipids have a natural tendency to form spherical shapes when placed in water, arranging themselves so that their hydrophobic tails point inward, away from the surrounding liquid, while their hydrophilic heads face outward. This tendency is not driven by intention but by physical laws, a gentle response to the properties of water and the structures of the molecules themselves. As lipids accumulated in the early ocean, they spontaneously formed small vesicles, tiny bubbles with watery interiors. These vesicles represented the first primitive boundaries on Earth, offering pockets of stability amid the fluid environment of the ocean. Within these vesicles, molecules became concentrated, trapped in close proximity. This was a significant change. Without a boundary, molecules diluted rapidly in the ocean, making interactions sporadic and unpredictable. But inside a lipid vesicle, the chances of interaction increased. A replicating molecule, once enclosed, no longer drifted freely into the open sea. It continued its copying process within a confined space, shaping the internal chemistry of its small environment. Vesicles containing replicating molecules grew more complex than their empty counterparts, becoming early prototypes of cells. These structures did not have the sophisticated machinery of modern organisms. No proteins, no true organelles, and no targeted functions. Yet they represented the first hints of individuality on Earth. Each vesicle carried its own internal mixture of molecules. Some of these vesicles would have been extraordinarily fragile. They could break apart under turbulent conditions or collapse when exposed to excessive heat. But others survived long enough for their internal chemistry to evolve. As replicating molecules inside the vesicles began interacting with other molecules, new reactions became possible. The presence of catalytic chains or strands that encouraged certain reactions over others allowed these early compartments to develop distinct characteristics. One vesicle might contain molecules that favored rapid replication, while another enclosed molecules that folded into stabilizing shapes. Over time, this diversity created a gentle selection process, where vesicles capable of maintaining their structure and supporting internal reactions persisted longer than those that could not. The oceans provided continuous support for these developments. Water served as a protective cushion, moderating temperature changes and shielding delicate structures from harsh radiation that would have quickly degraded them on land. Minerals dissolved in the seawater interacted with vesicles, sometimes stabilizing their membranes or participating in internal reactions. Hydrothermal vents offered energy-rich environments where vesicles could form, dissolve, and reform repeatedly, each cycle producing new combinations of internal molecules. These environmental factors did not choose structures intentionally, but created conditions where certain arrangements were more likely to survive. Through this gentle interplay between the environment and the emerging chemical systems, the earliest precursors to living cells began to take shape. As vesicles grew more stable, they occasionally merged with one another. When two lipid bubbles combined, their internal contents mixed, creating new combinations of molecules. Some of these combinations produced improvements, faster replication, more stable structures, or more efficient catalytic activity. Others produced no benefit or caused the vesicle to break apart. But over long periods, beneficial combinations became more common simply because they endured. Even without true heredity, a vesicle that remained intact and stable for longer periods allowed its internal molecules to replicate and influence future vesicles that formed from its remnants. Although this was not heredity as we understand it today, it represented continuity across generations of chemical structures. Inside these vesicles, another quiet transformation was beginning. Some molecules inside the compartments acted like early metabolic catalysts, facilitating reactions that transformed the surrounding molecules into new forms. These early metabolic networks were extremely simple compared to modern biochemical cycles, but they provided a foundation for more elaborate processes to develop. A vesicle capable of producing molecules that reinforced its own membrane, for example, had an advantage over those that did not. Over time, these self-reinforcing tendencies allowed some vesicles to persist in environments that would otherwise destroy them. The ability to regulate internal reactions, even in primitive ways, set the stage for the more sophisticated homeostasis seen in living organisms. Even the act of growing became possible in this environment. Vesicles could incorporate additional lipids from the surrounding water, expanding their membranes. As they grew, internal molecules continued replicating, filling the larger space. Eventually, vesicles became so large that their membranes destabilized and split into two smaller compartments. This process bears a striking resemblance to cell division, though it occurred without any of the regulatory mechanisms present in modern organisms. The splitting was solely driven by physical forces, yet it had profound consequences. When a vesicle divided, it passed its internal molecules to its daughter compartments. If those molecules included replicators or catalytic structures, then the new vesicles carried the same internal tendencies. With each generation, favorable molecular combinations became more widespread. Through these gentle and gradual processes, the line between chemistry and life began to blur. The planet had created environments where molecules could not only form, but also replicate, concentrate, evolve, and pass their characteristics forward. The earliest vesicles may not have been fully alive, but they embodied essential qualities that life would later refine. They protected their internal components, promoted interactions, and generated conditions where complexity could slowly accumulate. Most importantly, they represented individual units capable of variation and persistence, a necessary step in the long journey toward true cells. As millions of years passed, the primitive cells grew increasingly stable and complex. They developed more refined metabolic networks, more dependable replication strategies, and more robust membranes. Eventually, these early forms gave rise to the first true prokaryotic cells, organisms capable of independent survival and reproduction. But long before that transition occurred, the quiet emergence of vesicles marked a turning point. It was here, in these fragile bubbles drifting through the ancestral ocean, that life's first boundaries appeared soft, simple, and profoundly transformative. 6. Life becomes prokaryotic. In the slow unfolding of Earth's ancient story, after the first fragile compartments formed and molecules learned to copy themselves within protective boundaries, a new chapter began to take shape. This chapter would stretch across billions of years and lay the groundwork for all future organisms, from simple microbes to towering trees and thinking beings. It was the era in which the first true cells emerged, prokaryotes, the earliest ancestors of every living creature. These cells were small and structurally simple, yet they carried within them all the essential features of life. They could grow, respond to their environment, metabolize energy, reproduce, and evolve. In their quiet simplicity, they were the first undeniable living organisms on Earth, marking the transition from a world shaped only by chemical interactions to one shaped by biology. To picture these earliest prokaryotic cells clearly, it helps to envision them not as complex organisms but as delicate droplets of life drifting through the warm oceans. They resembled tiny spheres or rod-like shapes surrounded by thin membranes that separated their internal chemistry from the surrounding environment. Inside these membranes, molecules that once drifted freely in the ocean were now organized into purposeful systems. Genetic material, likely in the form of RNA at first, and later DNA, resided in the center, storing information that could be passed from one generation to the next. Surrounding it were simple metabolic pathways, networks of chemical reactions that allowed the cell to extract energy from its environment. Some prokaryotes used minerals, others relied on chemicals in the water. Still, others harnessed sunlight through primitive photosynthetic processes. This diversity in energy use demonstrated the adaptability that would become a hallmark of life. Prokaryotic cells also contained small structures known as ribosomes, essential for building proteins. These proteins acted as catalysts and structural components, enabling the cell to maintain its shape, perform chemical reactions, and respond to its environment. Though ribosomes lacked the complexity of later cellular machinery found in eukaryotic cells, they represented a significant leap in biological evolution. They allowed cells to translate genetic information into functional molecules, creating a direct link between heredity and metabolism. This connection became one of the defining features of life, an elegant system through which information shaped action and action influenced survival. These early prokaryotes were simple, yet they thrived in a variety of environments. Some lived near hydrothermal vents, where minerals and heat provided abundant energy. Others drifted through shallower waters, exposed to sunlight and circulating nutrients. Still others may have existed in sediments, interacting with the minerals of the ocean floor. Their small size allowed them to survive in conditions that would have been inhospitable to larger structures. They reproduced quickly, dividing through binary fission, a gentle process in which the cell grew, replicated its genetic material, and split into two nearly identical daughter cells. This ability to reproduce efficiently allowed them to fill the oceans, creating vast populations that would shape the chemical environment of the planet. Over time, prokaryotes diversified into two major groups, bacteria and archaea. Though both groups shared a simple structure, they differed in the composition of their cell membranes, their metabolic strategies, and their genetic machinery. Bacteria became especially common in environments ranging from shallow seas to deep sediments, where they played major roles in nutrient cycling. Archaea, by contrast, often thrived in extreme environments, hot springs, acidic pools, and oxygen-poor regions of the deep ocean. Their ability to survive in these harsh conditions demonstrated the remarkable resilience of early life. They adapted to environments shaped by volcanic activity, intense heat, or chemical extremes, developing specialized strategies that allowed them to persist where few others could. This resilience would eventually enable life to spread to every corner of the planet. One of the most profound contributions of prokaryotes to Earth's history came through the gradual development of photosynthesis. While early forms of photosynthesis may have been simple and inefficient, they paved the way for a remarkable transformation. Certain prokaryotes learned to use sunlight as a primary source of energy, capturing it through pigments and using it to convert carbon dioxide and water into organic compounds. At first, this process did not release oxygen, but eventually, a type of photosynthetic prokaryote, cyanobacteria, evolved a form of photosynthesis that did. With time, the oxygen produced by these cells accumulated in the atmosphere, fundamentally altering the composition of the air and the chemistry of the oceans. This rise in oxygen created new opportunities for life, allowing for more efficient energy production and setting the stage for the emergence of complex organisms. It also resulted in the Great Oxygenation Event, a quiet but monumental shift that changed Earth forever. As prokaryotes continued to evolve, they developed an impressive range of metabolic abilities. Some extracted energy from sulfur compounds, others from methane, hydrogen, or iron. This metabolic diversity allowed them to occupy nearly every environment the planet offered. They were not restricted by the narrow limits that larger, more complex organisms would eventually face. Instead, they flourished in hot, cold, acidic, alkaline, oxygen-rich, and oxygen-deprived environments alike. Their adaptability demonstrated that life does not require comfort, it simply requires opportunity. And with each passing generation, prokaryotes explored these opportunities, creating new niches and expanding their influence across the planet. Their interactions with the environment gradually shaped global cycles. They contributed to the formation of soils, influenced the composition of the atmosphere, and interacted with minerals in ways that transformed the surface of the Earth. In quiet and subtle ways, they acted as the planet's first engineers, shaping the world that future organisms would inherit. Their vast populations allowed them to evolve rapidly, adjusting to changes in climate, chemistry, and available resources. Even small evolutionary shifts could spread quickly through large populations, fostering innovation on a timescale far faster than what larger organisms could achieve. Although prokaryotes remained simple for billions of years, their simplicity should not be mistaken for insignificance. They established nearly all the foundational traits that life would later refine. They developed mechanisms for replication, energy use, and internal regulation. They formed communities, such as biofilms, that provided protection and stability. These communities hinted at the cooperative behaviors that would one day enable the rise of multicellular life. Prokaryotes also experienced genetic exchange, sharing fragments of genetic material through processes like horizontal gene transfer. This ability allowed them to acquire new traits rapidly, enhancing their adaptability and diversity. While modern organisms rely primarily on reproduction to pass traits forward, prokaryotes expanded their evolutionary toolkit by borrowing and exchanging genetic material, creating webs of shared information across populations. Through these simple yet powerful processes, prokaryotic life became not just a presence on Earth but a dominant force. They filled the oceans, covered the land masses once they formed, and inhabited environments previously thought uninhabitable. They set in motion the chemical changes that would allow more complex cells to arise. And though they remained microscopic, their influence stretched across every part of the planet, preparing the world for the next monumental shift. The rise of eukaryotic cells. In this gentle, gradual unfolding, prokaryotes established the principles of life with a quiet elegance. Their story is not one of dramatic leaps but of steady persistence and adaptation. Even today, billions of years later, prokaryotes continue to play essential roles in ecosystems, in the human body, and in the cycling of nutrients across the globe. They remind us that complexity does not always require grandeur. Sometimes it begins with the smallest of steps, taken softly in the ancient waters of a world just learning to breathe. 7. Photosynthesis and the slow rise of oxygen. In the gentle unfolding of Earth's early story, after the first prokaryotic cells took shape and settled into their quiet rhythms of survival, a new transformation began, one so gradual and so subtle that, for millions of years, the planet hardly noticed its presence. Yet this change would eventually alter the atmosphere, the oceans, and the destiny of every organism that would ever live. It began with a simple ability: the ability to capture light. In the calm shallows and drifting waters of the ancient seas, some of the earliest prokaryotes developed pigments capable of absorbing sunlight and converting it into energy. This was the beginning of photosynthesis, a process that would reshape the entire world in slow, steady waves. At first, photosynthesis did not produce oxygen. Early forms of the process used molecules such as hydrogen sulfide or other chemical donors to drive their reactions. These early phototrophic organisms harvested sunlight in a quiet, uncomplicated manner, storing energy in molecular bonds that helped them survive and divide. Their presence introduced a new kind of stability, allowing cells to rely on a consistent, abundant energy source instead of depending solely on chemicals drifting through the ocean. The sun, shining softly through the hazy atmosphere, became a gentle and reliable provider. With its light, organisms had access to a continuous supply of energy, slowly freeing life from the constraints of limited chemical environments. Over time, however, a group of prokaryotes evolved a more complex form of photosynthesis, one that used water as the electron donor. This change was profound, though it occurred without any awareness of its significance. When these ancient cyanobacteria absorbed sunlight and split water molecules to capture the needed electrons, oxygen was released as a byproduct. At first, this oxygen dissolved into the surrounding water, reacting with minerals such as iron. The iron in the oceans bound with oxygen, forming solid oxides that drifted softly to the seafloor. Layer by layer, these iron-rich sediments accumulated, creating banded iron formations that remain visible in rocks today. These layers are a record of the gradual rise of oxygen, quietly marking the shift from a world shaped entirely by geology and chemistry to one influenced by the metabolic activities of living cells. For a long time, the oxygen produced in this early photosynthesis was absorbed into the ocean or reacted with gases in the atmosphere. It did not accumulate in the air immediately. It took millions of years for the planet to become saturated enough for oxygen to begin rising into the atmosphere. But slowly, with each passing generation of cyanobacteria, more oxygen entered the environment. It is calming to imagine this transition not as an abrupt event, but as a gentle, continuous process. Day after day, sunlight fell upon the oceans. Day after day, photosynthetic cells drifted, absorbed light, and released oxygen. No organism understood the importance of what it was doing, yet the collective activity of these tiny, ancient cells gradually transformed the Earth. As oxygen levels increased, the oceans themselves began to change. Minerals settled differently. Chemical reactions shifted. Some organisms struggled to survive in the newly oxygenated waters. Previously stable molecules became reactive in the presence of oxygen, and many early anaerobic organisms, those that thrived without oxygen, found themselves restricted to isolated niches where oxygen could not penetrate. These shifts were not violent disruptions, but slow, persistent pressures that nudged life in new directions. Some organisms adapted, finding ways to tolerate oxygen. Others avoided it, and still others discovered how to use oxygen as a powerful tool, harnessing its reactive nature to produce energy more efficiently than ever before. The introduction of oxygen into Earth's atmosphere brought with it another gentle but transformative change: the formation of the ozone layer. When some of the newly released oxygen molecules drifted upward into the upper atmosphere, they were exposed to sunlight that broke them into individual atoms. These atoms combined with other oxygen molecules, forming ozone. Over long stretches of time, this layer thickened into a protective shield that softened the harsh ultraviolet radiation reaching the planet's surface. With this protection, future life would be able to explore new environments, including shallow waters and, much later, the land. Without the ozone formed by oxygen, the delicate molecules needed for life's growth would have been too easily damaged by solar radiation. Thus, through a slow and unplanned process, photosynthetic microbes created conditions that made the future diversity of life possible. Despite these overwhelming planetary changes, the lives of individual photosynthetic prokaryotes remained simple and serene. They floated gently through the water, following currents, responding to light, dividing softly into new cells. They formed mats and colonies on rocks and sediments, creating textured layers known as stromatolites, structures still found in isolated places on Earth today. These stromatolites offer a window into the past, showing how ancient communities of photosynthetic cells shaped their surroundings, layer by layer. As cells grew, divided, and became buried by new generations, they built structures that recorded the rhythms of their existence. These microbial communities were among the earliest examples of cooperative living systems, hinting at the more complex organizations that would later arise. The slow rise of oxygen also transformed the atmosphere. As oxygen accumulated in the air, the sky gradually shifted toward the familiar blue we recognize today. The increase in atmospheric oxygen introduced new chemical processes, helping break down gases like methane that once warmed the Earth. This change may have contributed to periods of cooling, including ancient glaciations that tested the resilience of early life. Yet even through these changes, photosynthetic organisms continued their quiet work. Their persistence ensured that oxygen levels remained stable enough to support future innovations in metabolism and cellular structure. As oxygen became more abundant, some prokaryotes developed the ability to use it directly in their energy production. This development, cellular respiration, allowed organisms to extract far more energy from organic molecules than earlier metabolic pathways could provide. Oxygen, with its strong tendency to react, offered a highly efficient means of releasing energy from food sources. This increase in available energy opened the door for larger and more complex cells to evolve, setting the stage for the later rise of eukaryotic organisms. In this way, the slow rise of oxygen gently guided life toward greater complexity, offering opportunities that would not have been possible in an anoxic world. And yet, despite its monumental impact, the emergence of photosynthesis remained a quiet affair. There were no sudden leaps, no rapid bursts of change, only the steady, rhythmic work of countless microscopic cells drifting in the oceans, capturing sunlight and releasing oxygen. Their existence was simple, but their influence was immeasurable. They taught the planet how to breathe. They created the air that would one day support forests, animals, and human beings. They shaped the chemistry of the oceans and prepared the world for life's next great transformations. In the gentle stillness of those early ages, photosynthesis offered a new kind of stability, a new source of energy, and a new path forward. It illuminated the oceans with invisible potential, encouraging life to explore new forms and new strategies. Through billions of tiny actions, each quiet and unrecognized, the story of Earth turned toward complexity, and the planet, once shaped only by stone and water, began to glow with the soft breath of living things. 8. Life learns to cooperate. The power of symbiosis. In the unfolding story of early life on Earth, after photosynthesis had begun to slowly fill the atmosphere with oxygen, and prokaryotic cells had settled into their ancient rhythms, a quiet and profound change began to take shape. Instead, it emerged gradually through countless interactions between tiny cells drifting in the primordial oceans. It was here, in these calm waters, that life discovered one of its most enduring strategies: cooperation. And from this cooperation, a new form of complexity began to grow, complexity that would eventually lead to the rise of eukaryotic cells, multicellular organisms, and all of the intricate living structures that would come to populate the planet. To imagine this transition, it is helpful to see the early ocean as a world filled with simple cells, each striving in its own way to survive. Most were solitary organisms, carrying only the minimal systems needed to gather energy, replicate. They floated gently through the water, dividing when conditions allowed and resting when resources grew scarce. But as the presence of oxygen increased, the oceans began to shift. Some cells struggled to cope with the reactive nature of oxygen, which could damage their internal structures. Others adapted to tolerate it, and a rare few learned to use oxygen as a tool, harnessing its energetic potential through respiration. These new oxygen-using cells could extract far more energy from their environment than their anaerobic neighbors, granting them increased endurance and stability. It was in this shifting landscape that cooperation emerged as a quiet force of evolution. Over time, some prokaryotic cells found themselves living in close proximity to others. Perhaps they drifted together in protective mats, or perhaps they became trapped within mineral compartments or lipid vesicles. In some cases, one cell entered another by accident, slipping through a weakened membrane, or becoming engulfed through a process resembling primitive ingestion. Whatever the circumstances, the result was extraordinary. Certain engulfed cells could detoxify oxygen, allowing the host to survive in an increasingly oxygen-rich world. Others produced energy more efficiently, sharing this energy with their host cell. Over time, these mutually beneficial relationships deepened, growing into true symbiosis. In this gentle partnership, the engulfed cells lost their independence gradually, transferring portions of their genetic material to their host. In return, the host provided protection, stability, and a reliable internal environment. This exchange, subtle and incremental, paved the way for the formation of mitochondria organelles that would become essential to nearly all eukaryotic life. The host cell gained access to efficient energy production, while the engulfed cell found a sheltered home. Neither organism planned this relationship, and neither understood the transformation that was unfolding. But through the slow rhythm of generations, through survival and persistence, the partnership deepened into a permanent arrangement. The origins of mitochondria represent one of the greatest examples of cooperation in Earth's history. Another symbiotic partnership arose between ancient cells that could perform photosynthesis and their hosts. These engulfed cells, similar to modern cyanobacteria, brought with them the ability to capture sunlight and produce organic molecules. Over time, they evolved into chloroplasts, the organelles that allow plants and algae to carry out photosynthesis today. Like mitochondria, chloroplasts bear traces of their prokaryotic ancestry, including their own DNA. These clues, preserved across billions of years, remind us that complexity often begins with cooperation. By working together, one cell providing energy, the other offering protection, life discovered new forms and new possibilities. The emergence of these symbiotic relationships marked a turning point. Cells that had once survived on limited chemical resources could now generate abundant energy. This surplus allowed them to develop more intricate internal structures. They formed cytoskeletons that offered shape and movement. They developed systems to transport molecules within their membranes. Their genetic material became organized inside a nucleus, allowing more refined control over gene expression. Bit by bit, they grew into the first eukaryotic cells, cells far larger and more complex than their prokaryotic ancestors. These new cells represented an evolutionary leap, but they were built upon the quiet cooperation of simpler forms. Though this transformation was profound, it is soothing to imagine it unfolding not as a dramatic breakthrough, but as a slow, gentle drift toward complexity. Over millions of years, host cells and their internal partners adapted to one another. The engulfed cells gradually relinquished their autonomy, shedding genes that were no longer needed. The host cells, now relying on their symbiotic partners, developed systems to manage and support them. This cooperative integration created a new kind of life, one capable of greater flexibility, endurance, and variety than the world had ever seen. Beyond the formation of organelles, symbiosis also shaped the behaviors of early life. Some cells began forming colonies, where individual members worked together in loose arrangements. These colonies offered protection from environmental stress, improved access to nutrients, and increased chances of survival. Though simple, they represented early versions of communal living. Over time, colonies became more organized. Cells began taking on specialized roles, performing tasks that benefited the group rather than themselves alone. These early forms of cooperation foreshadowed the rise of true multicellularity, where cells acted not as individuals but as harmonious parts of a larger organism. In every corner of the early ocean, cooperation emerged as a quiet, guiding force. It did not eliminate competition or replace the struggle for survival, but it offered another path, one in which organisms could thrive through mutual support. The story of life's early evolution is often told as a tale of adaptation and survival, but it is also a story of partnership. Mitochondria and chloroplasts represent living evidence of how powerful cooperation can be, even at the smallest scale. They continue to function in our own cells today, quietly producing energy and sustaining the processes that allow us to live, breathe, and think. As these symbiotic relationships strengthened, the planet grew more prepared for the next steps in the evolution of complexity. The oceans, once populated only by simple prokaryotes, now contained larger, more versatile cells capable of exploring new environments and forming new communities. These eukaryotic cells laid the foundation for multicellular organisms, ecosystems, and the vast diversity of life that now covers the Earth. But at the heart of it all, beneath the intricate layers of biological design, rests the simple truth that life's greatest innovations often begin with cooperation. In the gentle currents of the ancient seas, two cells, finding harmony within one another, set into motion a cascade of changes that would shape all living things. Through patience, persistence, and the quiet elegance of symbiosis, life learned that it could become more by working together than it ever could alone. 9. The arrival of eukaryotic cells. In the quiet expanse of Earth's early oceans, after cooperation had become a subtle guiding force, and simple prokaryotic cells had learned to coexist in mutually beneficial ways, a new form of life began to unfold. This transformation did not occur suddenly or with sharp boundaries. Instead, it emerged slowly, layer by gentle layer, as early cells experimented with new structures, new forms of organization, and new ways of storing and using energy. The world was still dominated by the simplicity of prokaryotes, but within certain compartments of the ocean, something different was arising: cells with internal divisions, cells with protected genetic material, cells that carried within them the echoes of ancient partnerships. These were the first eukaryotic cells, and their appearance marked one of the most profound transitions in life's long history. To imagine the first eukaryotic cells, It may help to visualize them as gently expanding spheres, larger and more complex than their prokaryotic ancestors. Within these early eukaryotes was a defining feature: the nucleus, a specialized compartment enclosed by a membrane. Inside this nucleus, genetic material found a new level of protection and organization. DNA, once floating freely in the cytoplasm of simpler cells, was now stored safely within this membrane, shielded from harmful reactions occurring elsewhere in the cell. This separation allowed the genetic material to replicate more accurately and to be expressed in more refined ways. The nucleus did not arise through sudden inspiration, but through gradual shifts in membrane behavior and internal structure, shaped by the quiet persistence of evolutionary time. Surrounding the nucleus, the cytoplasm of eukaryotic cells carried a growing collection of organelles. These organelles, each enclosed in their own membranes, performed tasks that earlier cells had accomplished only through simpler, less coordinated means. Mitochondria, once independent prokaryotes, now powered the cell with their ability to harness oxygen efficiently. Their presence allowed eukaryotic cells to sustain far larger sizes and more demanding processes than prokaryotes ever could. With access to the potent energy produced through aerobic respiration, eukaryotes began exploring new pathways of growth and interaction. They were no longer limited by the constraints of low-efficiency metabolism. Instead, they became capable of supporting intricate internal networks, each contributing to a stable and self-sustaining whole. Other organelles emerged through similar processes of internal evolution and membrane enfolding. The endoplasmic reticulum provided surfaces for building proteins and lipids, while the Golgi apparatus refined and packaged these molecules for specific uses. Vacuoles and vesicles helped store materials or transport them through the cell's interior. Lysosomes offered methods for breaking down and recycling cellular components. Each new structure added another layer of organization, allowing eukaryotic cells to perform tasks previously impossible. Though these organelles varied in origin and function, they shared a common purpose: to support a more complex and adaptable form of life. The cytoskeleton, a dynamic network of protein filaments, offered structural support and mobility. In prokaryotic cells, shape was often determined by simple molecular patterns. But in eukaryotes, the cytoskeleton allowed cells to alter their form, extend pseudopods, and move with fluid grace. It also provided a framework for transporting materials internally, guiding molecules to where they were needed and ensuring that cellular processes unfolded smoothly. This internal scaffolding represented a significant step toward the ability to coordinate tasks across different parts of the cell. It brought a sense of internal organization reminiscent of a small, self-contained world, a world shaped not by external currents, but by its own internal rhythm. The emergence of eukaryotic cells also brought a new sophistication to reproduction. Instead of relying solely on binary fission, early eukaryotes developed forms of cell division that allowed for more accurate distribution of genetic material. Mitosis ensured that DNA was carefully replicated and divided, reducing errors and preserving the integrity of the cell's information. This fidelity allowed eukaryotic populations to maintain stability across generations, even as they explored new environments and conditions. Later, the evolution of sexual reproduction introduced a new source of variation, though this innovation lay far ahead in time. For now, the quiet division of early eukaryotes was enough to sustain their growing presence in the oceans. One of the remarkable aspects of early eukaryotic evolution was its adaptability. Eukaryotic cells could grow larger than prokaryotes, giving them access to new ecological niches. Their internal complexity allowed them to store nutrients, respond to environmental changes with greater sensitivity, and perform multiple tasks at once. These abilities opened opportunities that had not existed before. Some eukaryotes drifted through the oceans as solitary cells, while others formed simple colonies, beginning the long journey toward multicellularity. The increased size and complexity of eukaryotes also made predation feasible, a gentle precursor to future ecological interactions. As certain cells developed feeding structures capable of engulfing smaller organisms, interactions between species grew more dynamic, adding depth to the ecological landscape of the ancient oceans. The diversity of eukaryotes expanded gradually. Some became ancestors of modern algae, capturing sunlight and filling the oceans with green, gold, and red hues. Others became ancestors of fungi, developing metabolic pathways suited to absorbing nutrients from their surroundings. Still others evolved into protozoa, moving through the water with graceful motions powered by cilia or flagella. These early eukaryotic lineages experimented with countless forms and strategies, each shaped by the interplay of environmental pressures and internal innovation. Their existence added new layers of complexity to the planet's ecosystems, influencing not just the chemistry of the oceans but the flow of energy and the cycling of nutrients. Despite their increasing complexity, the lives of early eukaryotes were still serene. They floated through the water, responding gently to currents and light, dividing when conditions allowed, and resting when resources were scarce. Their organelles functioned quietly inside them, performing countless tasks without fanfare. The gradual accumulation of these functions created a level of organization that far surpassed anything Earth had seen before. Yet this complexity did not disrupt the rhythm of the early world. Instead, it became part of the gentle continuity of life, woven into the steady progression of time and change. The arrival of eukaryotic cells prepared the planet for everything that would follow. Without their internal organization, multicellularity could not have evolved. Without mitochondria, energy-intensive processes such as movement, communication, and coordinated growth would have been impossible. Without a nucleus to safeguard genetic material, the vast diversity of life that would one day emerge from forests to animals to humans could not have arisen. These early eukaryotes were the quiet architects of the future, carrying within them the potential for all the complexity life would later express. As we pause at this point in the story, it becomes clear that life did not leap forward suddenly. Instead, it advanced through small, cumulative steps, each one subtle, each one adding a new thread to the fabric of biological possibility. The first eukaryotic cells represent a turning point in this tapestry, a blending of cooperation, structure, and stability that transformed the oceans forever. From their gentle beginnings, the story of life's increasing intricacy moved forward, preparing the way for multicellular forms, ecosystems, and the rich diversity of the living world. 10. Multicellularity begins cells join in community. In the long, gentle unfolding of Earth's biological story, after the rise of eukaryotic cells brought new levels of internal organization and quiet sophistication, the next great transition began in the stillness of the ancient seas. It was a transition not born from sudden innovation, but from subtle shifts that took place over millions of years. Simple, solitary cells, content to drift alone through the water, gradually discovered the quiet strength of companionship. This was the beginning of multicellularity, a step that would eventually lead to forests, coral reefs, animals, and every complex organism that would one day walk or swim or grow across the planet. But at first, it began softly, as life often does, with small groups of cells finding comfort and advantage in living together rather than apart. It is soothing to imagine these early eukaryotic cells gently floating in the oceans, each carrying within it the inherited complexity of its internal structures. They possessed nuclei to protect their genetic information, mitochondria to provide efficient energy, and membranes capable of forming delicate shapes that allowed them to interact with their surroundings. These abilities gave them resilience and adaptability, allowing them to thrive in environments that were inaccessible to simpler prokaryotes. Yet even with these advantages, they remained solitary beings for a very long time. The world around them, though stable in many ways, was full of unpredictable currents. shifting nutrient levels, and varying conditions that tested their endurance. In this fluctuating environment, small clusters of cells occasionally remained together after division, forming simple aggregates. At first, these clusters were temporary, just brief moments when daughter cells drifted side by side before separating. But sometimes the cells remained connected through thin bridges of cytoplasm or shared extracellular matrix material. These subtle connections offered the first hints of communal living. Within these groups, cells benefited from being in close proximity. They were less vulnerable to predation, as clusters could appear larger and therefore more difficult to engulf. They could also share resources more effectively, passing molecules from one cell to another or collectively responding to environmental changes. In a world governed by gradual pressures, even these small advantages mattered. As time moved forward, these temporary groupings became more stable. Some cells evolved the ability to secrete sticky substances that held them together. Others began to divide in ways that naturally maintained proximity, forming chains, sheets, or small spherical colonies. These early multicellular structures were simple and lacked the tight coordination seen in later organisms, but they represented significant progress. They marked the moment when life began to explore the possibilities of cooperation, not just between unrelated organisms, as seen in symbiosis, but between related cells sharing the same genetic blueprint. Over countless generations, the cells within these clusters began to adopt different behaviors. Some focused on capturing sunlight, drifting near the surface where light was more abundant. Others became better at absorbing nutrients or expelling waste. These differences were not yet true specialization, but they hinted at what was to come. The fact that some cells could take on slightly different roles allowed the entire group to function more effectively than any single cell could alone. This subtle division of labor laid the earliest foundations for tissues and organs. The environment continued to play a supportive role in this shift. Calm, shallow waters allowed colonies to grow without being torn apart by strong currents. Sediment layers created sheltered pockets where cells could remain together longer. Nutrient gradients encouraged cells to stay in regions where conditions were favorable. These gentle influences encouraged multicellular clusters to persist and expand, gradually becoming more intricate. The presence of predators also contributed. As predatory eukaryotes became more common, solitary cells faced increasing risk, while groups had a better chance of survival simply through numbers. Through slow, persistent pressure, survival favored those cells that could remain connected and coordinate their efforts. One of the most important developments during this period was the emergence of communication between cells. Though primitive, early cells could send simple chemical signals to their neighbors. These signals helped synchronize activities, allowing groups to contract or expand, to move as a unit, or to respond collectively to changes in their environment. This communication strengthened the cohesiveness of the group, making separation less likely and cooperation more beneficial. Over time, these signals evolved into more refined systems, supporting the coordination required for more complex multicellular life. Another gentle transition unfolded as some cell clusters began showing consistent patterns of arrangement. Instead of forming random shapes, some groups developed symmetry or repeated geometric patterns. These patterns allowed the colony to function more efficiently. For example, cells positioned on the outer surface of a sphere might become more involved in interacting with the environment, while inner cells focused on protection or nutrient storage. This layering represented an early form of structural organization, one that would eventually lead to specialized tissues. As multicellular structures became more established, the cells within them began experiencing different environments, depending on their position. Cells on the outside faced the ocean directly, feeling the flow of water and access to nutrients. Inner cells experienced more stability, but were more dependent on their neighbors for resources. These differences gradually drove the development of cellular specialization, Some cells became better suited to absorbing nutrients, while others grew more efficient at passing them inward. Some cells produced structural materials to strengthen the colony, while others focused on reproduction. In this way, differentiation, one of the cornerstones of complex life, emerged gently, with no single moment marking its beginning. Rather, it evolved through small adjustments that accumulated across countless generations. The development of multicellularity also expanded the ways in which life interacted with its environment. Larger organisms could access deeper layers of water or attach to surfaces. They could move as coordinated units or float more efficiently. These advantages allowed multicellular organisms to explore and thrive in environments that had previously been inaccessible. Even with these emerging complexities, early multicellular life remained peaceful and unhurried. There were no rapid leaps toward the forms we recognize today. Instead, there was a long, gradual unfolding of possibilities. Colonies gently added layers of cells. Patterns slowly became more regular. Interactions took on new depths of subtlety. And as eukaryotic life continued this quiet journey, the blueprint for more intricate organisms began to take shape. The birth of multicellularity was not a single event, but a continuing process, one that would shape the entire future of the planet. In these early ages, the ocean became home not just to solitary cells, but to small communities of cooperating beings. They were the first to discover that survival could be shared, that actions could be coordinated, and that the future could be built together rather than alone. 11. The emergence of tissues. A gentle step toward organization. In the soft progression of life's early evolution, after simple organisms learned to live together in colonies, and the first hints of multicellularity began to unfold, the planet quietly prepared for another profound yet gentle transition. This transition did not announce itself with sudden complexity or dramatic forms. Instead, it emerged slowly, almost imperceptibly, as cells within these early clusters began to take on more defined roles. From this subtle shift arose one of life's most elegant innovations: tissues. These early tissues were not yet the intricate structures seen in modern organisms, but they represented the moment when cells began working in harmony toward specific functions, moving life one step closer to organized multicellularity. To imagine this stage clearly, visualize the ancient ocean filled with floating colonies of cells, simple spheres, sheets, or filaments, each composed of related cells that had chosen to remain together after division. As these organisms drifted through the waters, their layered arrangements exposed certain cells to different surroundings than others. Cells on the outside felt the currents of the ocean, experienced direct sunlight, and absorbed nutrients first. Meanwhile, cells buried deeper within the clusters were sheltered from environmental changes but depended heavily on their neighbors for resources and protection. This natural gradient of experiences gently nudged cells into performing different roles based on where they were located. At first, these differences were minimal. Surface cells might have become slightly flatter or better suited to filtering nutrients, while interior cells became more rounded and supportive. There was no formal system of specialization, only small variations shaped by necessity. But as time passed, these subtle differences began to strengthen. With each generation, the offspring of surface cells carried tendencies that made them more efficient at interacting with the environment, while the offspring of interior cells became more effective at storing nutrients or providing structural support. Across countless cycles of growth, division, and adaptation, these tendencies developed into early forms of specialization, cells performing specific tasks not for themselves alone, but for the benefit of the entire group. These gentle beginnings of specialization laid the foundation for tissues. A tissue, in its simplest form, is a group of similar cells working together to carry out a shared function. In these early organisms, the first tissues emerged as layered arrangements of cells that had committed to particular tasks. The outermost cells gradually developed into protective layers, Their membranes thickened and their shapes flattened to create a barrier between the organism and the environment. These early protective tissues served roles that would one day be performed by skin or epithelial layers, shielding the internal cells from physical stress and regulating the flow of nutrients and waste. Within these protective layers, other cells began forming supportive networks. They became elongated or fibrous, creating a scaffold that helped maintain the shape of the organism. Though primitive, these supportive tissues offered stability, making the organism more resilient to the movement of water or the shifting sands of the ocean floor. Over time, as these supportive cells developed connections to one another, the group of cells moved together more gracefully. Their structure became more coordinated, allowing the cluster to grow larger without collapsing under its own weight. Another quiet transformation occurred when certain cells within the developing tissues began to specialize in communication. They sensed changes in light, temperature, or nutrient levels and conveyed these signals to other parts of the organism. This early form of cellular communication helped coordinate movements, growth, and responses to environmental changes. Though far simpler than the nervous or hormonal systems that would develop in later organisms, these first communicative tissues allowed multicellular life to act more cohesively, laying the groundwork for more advanced regulation. The emergence of tissues also encouraged organisms to explore new shapes. Instead of remaining as simple spheres or filaments, some organisms developed flattened bodies that increased their surface area, allowing for more efficient nutrient absorption. Others developed hollow centers, creating internal chambers where specialized cells could carry out tasks like digestion or reproduction more effectively. Each new structure represented an experiment, a gentle exploration of what multicellular organization could achieve. These shapes were not yet complex organs, but they hinted at the possibilities of organized internal environments, spaces where different tissues could perform coordinated functions. As tissues became more established, organisms also began showing early signs of directional growth. Some cells elongated, forming structures that resembled primitive stems or fronds. These growth patterns allowed the organisms to anchor themselves in place or extend into regions with better access to light or nutrients. The tissues at the tips of these structures often contained cells capable of rapid division, supporting continued expansion. This gentle extension into the environment represented a deeper level of interaction between organisms and their surroundings, moving life closer to the adaptive sophistication that would define future plant and animal lineages. The development of tissues also introduced new possibilities for reproduction. In simpler multicellular organisms, reproduction often involved breaking off a portion of the colony, which then continued to grow independently. But as tissues formed, some groups of cells became better suited to producing reproductive cells. These reproductive tissues gradually emerged as clusters of cells dedicated to creating new individuals. Their specialization allowed the organism to reproduce more efficiently and with greater control. Over time, this increased reproductive stability contributed to the success and spread of tissue-based organisms. Throughout this long transition, the early ocean remained a calm and supportive environment. The water's regulated temperature provided abundant minerals and protected delicate tissues from harsh conditions. Sunlight filtering through the surface allowed photosynthetic tissues to flourish, while deeper waters provided refuge for organisms sensitive to light. Through gentle waves and currents, the ocean carried nutrients to stationary organisms and transported reproductive cells across great distances. In this nurturing environment, tissues continued to refine their roles, becoming more structured and more coordinated. The emergence of tissues marked a major step toward complexity, yet it unfolded with remarkable tranquility. There were no abrupt leaps or sudden transformations, only the quiet accumulation of advantages gained through cooperation and specialization. With each small improvement, organisms became more resilient, more adaptive, and more capable of exploring new ecological possibilities. These early tissues laid the foundation for the organs and systems that would one day support breathing. But at this early stage, They served simple and essential roles: protecting, supporting, sensing, and sustaining the organism. As we reflect on this chapter in life's history, it becomes clear that tissues represent the harmony of cooperation at a cellular level. They are reminders that complexity grows from unity, from cells choosing to share tasks and responsibilities. In these ancient oceans, where the first tissues took shape, life began to organize itself in ways that would echo across all future lineages. 12. Organs and organ systems take shape. In the gentle progression of life's increasing organization, after tissues began forming simple layers and performing shared tasks, the ancient oceans quietly prepared the way for the next great emergence. This new step did not appear suddenly, nor did it arrive fully formed. Instead, it arose from the same slow, patient unfolding that had guided all earlier transitions. As tissues refined their functions and began cooperating with greater consistency, they gradually assembled themselves into larger, more intricate structures. These structures, capable of coordinating multiple tasks at once, would become the first organs, soft, functional arrangements that marked a new level of biological harmony. From these early organs, the seeds of organ systems began to sprout, creating networks of interaction that would one day support the bodies of plants, animals, fungi, and many other forms of life. To envision these early organs, imagine simple multicellular organisms drifting through calm seas, their tissues beginning to specialize in more distinct ways. Outer layers protected the organism, absorbing nutrients or capturing sunlight. Inner layers stored energy or processed materials. As these tissues grew more coordinated, some regions of the organism naturally took on roles that supported the needs of the whole. A thickened patch of tissue might become particularly good at absorbing nutrients. Another region might retain water or serve as a reservoir of minerals. These early specializations formed the prototypes for organs regions where cells worked collectively toward a single, essential purpose. The journey from tissue to organ was a delicate one, unfolding gradually across countless generations. Organisms did not suddenly possess hearts, lungs, or digestive tracts. Instead, they developed simpler biological structures that performed fundamental tasks. In early photosynthetic life, clusters of pigment-rich cells formed primitive light-harvesting organs, enhancing the organism's ability to capture sunlight. These structures were precursors to the leaf-like surfaces that plants would later develop. In other organisms, dense aggregations of cells acted as rudimentary digestive tissues, enclosing spaces where captured nutrients could be broken down more efficiently. These primitive organs improved the organism's ability to extract energy from its surroundings, fostering growth and resilience. As organs became more defined, interactions between them began to emerge. This marked the earliest formation of organ systems, groups of organs working together to support the entire organism. In simple multicellular forms, these systems were not complex or sharply differentiated, but they represented the beginning of coordinated biology. For example, tissues involved in nutrient absorption worked alongside tissues that distributed those nutrients through the organism's body. Supportive tissues strengthened outer layers, making it easier for nutrient-absorbing regions to remain exposed to favorable conditions without compromising structure. In this way, organisms began to rely on interdependent networks. The development of primitive circulatory systems also began during this time, though they bore little resemblance to the intricate systems of later animals. Instead of blood vessels or pumping structures, early organisms used diffusion and simple currents of fluid to move nutrients, gases, and waste products through their bodies. As organisms grew larger, diffusion alone became insufficient, leading to the gradual evolution of more directed internal pathways. These pathways allowed materials to move more efficiently, giving rise to internal channels that foreshadowed veins, arteries, and lymphatic systems. Though rudimentary, They represented the first stirring of organized internal transport. Meanwhile, structural organs grew increasingly refined. In some organisms, layers of supportive tissue thickened into stiffened frameworks that enabled upright growth or anchoring to surfaces. These frameworks were the early forms of what would eventually become stems, trunks, or skeletal systems. They provided stability, allowing the organism to grow taller or expand outward. The emergence of structural organs demonstrated how specialization fostered new physical forms, expanding the possibilities for life's architecture. As organs continued to diversify, reproductive structures also began to emerge. In early multicellular organisms, reproductive cells were often identical to other cells but located in specific regions. Over time, these regions became more specialized, forming tissues dedicated to producing new individuals. These early reproductive tissues were the blueprint for the reproductive organs that would appear later in plants, fungi, and animals. Their formation represented a shift toward more reliable and efficient reproduction, allowing organisms to pass on their traits with increasing consistency. The coordination of organ systems required new forms of communication. Earlier tissues had relied on simple chemical signals, but as organisms grew more complex, these signals became more nuanced. Cells communicated across greater distances, guiding growth, triggering responses to environmental changes, and maintaining internal balance. These signaling pathways did not yet resemble the hormonal or nervous systems of later life, but they laid the groundwork for internal regulation, a key feature of advanced multicellular organisms. Throughout these transitions, the environment continued to shape the evolution of organs and organ systems. The oceans provided gentle currents that carried nutrients and removed waste. Gradients of light and temperature encouraged certain organs to develop near the surface, while others specialized in functions deeper within the organism. Predatory pressures encouraged the formation of protective structures, while resource competition inspired adaptations that enhanced efficiency. Every environmental influence served as a quiet sculptor, guiding the direction of complexity in a patient and understated manner. As time passed, early multicellular organisms began exploring new shapes and lifestyles. Some grew flatter to maximize exposure to sunlight. Others formed tubular bodies that allowed water to pass through them, delivering nutrients directly to internal tissues. Some developed branching patterns, with repeated units of organs and tissues that could grow indefinitely in multiple directions. These shapes hinted at the modularity that would later characterize many plants and fungi. They also showed how organs could be arranged in repeating systems, supporting growth and survival in diverse conditions. The emergence of organs also shifted the relationship between organisms and their internal environments. Instead of simply reacting to external changes, organisms now managed internal processes through coordinated networks of tissues and organs. This internal control allowed them to maintain stable conditions even in fluctuating surroundings. In this sense, organs and organ systems represented a deeper level of biological independence, a way for life to shape itself from within, guided by gentle regulation and interwoven functionality. Looking back at this stage in Earth's story, it becomes clear that the rise of organs and organ systems marked a quiet ascent into greater complexity. It was a time when cells learned not only to cooperate but to specialize in ways that benefited the entire organism. It was a time when multicellular life took its first steps toward the structured bodies that would one day fill oceans, land, and air. And yet, through all these developments, the process remained gentle, steady, and beautifully unhurried. 13. Ecosystems form and interact, the first webs of life. As organs and organ systems began to take shape in the ancient seas, life continued its gentle expansion into new realms of complexity. Individual organisms, now equipped with specialized tissues and coordinated internal functions, were better able to interact with the world around them. They grew larger, more diverse. Yet, even as organisms evolved in their own quiet ways, another transformation unfolded not within individual bodies, but in the relationships between them. This transformation marked the emergence of ecosystems, interconnected communities of living beings and their physical environments, woven together in delicate networks of energy and exchange. These early ecosystems were simple compared to the forests, reefs, and grasslands that would come later, but they represented the beginning of ecological organization, a step in which life learned not only how to exist. To picture these early ecosystems, imagine the ancient oceans as expansive worlds filled with drifting organisms of all shapes and sizes. Some floated near the surface, capturing sunlight and producing energy-rich molecules. Others grazed softly upon these producers, absorbing nutrients and releasing waste that, in turn, nourished different organisms. Tiny predatory cells swam with gentle motion, feeding on smaller organisms and influencing population sizes. The ocean floor hosted communities that relied on minerals and thermal energy rather than sunlight, creating pockets of life that thrived independent of surface conditions. These interactions formed the earliest ecological webs, simple cycles of energy transfer and nutrient flow that shaped the environments they occupied. At the heart of these early ecosystems were producers, organisms capable of converting sunlight or chemical energy into organic matter. Cyanobacteria and early algae, with their photosynthetic machinery, provided a steady supply of energy to the surrounding life forms. They used light to generate nutrients not only for themselves, but inadvertently for countless other organisms. Their gentle activity helped stabilize the chemical balance of the oceans and introduced a regular influx of oxygen, slowly transforming the atmosphere and enabling new metabolic strategies. These producers served as the foundational layer of early ecosystems, supporting consumers and decomposers in a balanced, interconnected cycle. Consumers emerged from the growing diversity of eukaryotic life. Some were simple, free-floating grazers that fed on producers by engulfing them or absorbing their nutrients. Others developed more specialized feeding structures, allowing them to pursue specific types of prey or extract resources from particular environments. These early consumers introduced a new form of ecological interaction predation, which gently influenced the abundance of different organisms. Predation in these ancient seas was not violent in the way we might imagine today. It was a slow and natural part of the balance, with populations rising and falling based on the availability of food and the presence of predators. Through these interactions, ecosystems developed a quiet rhythm, a steady pulse of growth, decline, and renewal. Decomposers also played a vital role in maintaining balance. When organisms died, their bodies drifted downward, eventually settling on the ocean floor. Here, bacteria and other simple organisms broke down the organic matter, releasing nutrients back into the environment. These nutrients enriched the sediments and waters, supporting new life and completing the ecological cycle. This gentle recycling process ensured that nothing was wasted, that every molecule could return to the flow of life. Decomposers form the subtle but essential foundation of nutrient cycles, allowing ecosystems to endure and evolve. As interactions between producers, consumers, and decomposers grew more refined, ecosystems expanded in diversity. New forms of cooperation emerged. Some organisms offered shelter to others, providing surfaces on which they could grow or spaces in which they could reside. Others formed symbiotic relationships, exchanging resources or protection in ways that benefited both partners. These early forms of cooperation added layers of stability to ecosystems, allowing them to withstand changes in temperature, light, and nutrient availability. With each new relationship, life strengthened its connection to the environment and to itself, weaving an increasingly complex web. Energy flow became a defining feature of ecosystems. Sunlight entered the system through producers, traveled through consumers, and returned to the environment through decomposers. This flow of energy supported the structure of the system, determining how many organisms could survive at each level. Ecologists today describe this structure using trophic levels, But in these early ages, it was simply a natural balance maintained by the quiet interactions of living beings. Nutrient cycles also developed, with elements like carbon, nitrogen, and phosphorus moving between organisms and their environments. These cycles created a steady rhythm of transformation, allowing ecosystems to function smoothly over long periods. Habitats within these ecosystems varied widely. Near the ocean's surface, sunlight supported photosynthesis, creating vibrant communities of producers and small grazers. In deeper waters, dim light offered a gentler environment where organisms drifted slowly, relying on falling organic matter for nourishment. Near hydrothermal vents, ecosystems formed around chemical energy, supporting unique communities that operated independently of sunlight. These pockets of life demonstrated that ecosystems can form wherever stable sources of energy exist, whether from the sun, the Earth's interior, or the quiet interplay of nutrients. As ecosystems became more established, new forms of adaptation emerged. Organisms developed behaviors and structures that suited their ecological roles. Producers optimized their ability to capture light by expanding their surfaces or developing pigments suited to different wavelengths. Consumers evolved more efficient ways to locate food, avoid predators, or navigate the currents. Decomposers became increasingly effective at breaking down complex molecules, helping to maintain the nutrient balance of their environments. Through these adaptations, ecosystems grew more resilient, capable of sustaining life through subtle shifts in climate or chemical composition. These early ecosystems also fostered biodiversity, as different organisms adapted to specific ecological niches. Their populations diversified. Some explored the surface waters, others the mid-depths, and still others the ocean floor. This diversification created new interactions, new dependencies, and new opportunities for life to evolve. Biodiversity enriched ecosystems by providing stability. If one species declined, others could fill its role, ensuring that the flow of energy and nutrients continued uninterrupted. The variety of life forms also encouraged further evolution, as organisms adapted to coexist, compete, or cooperate with their neighbors. In these quiet ages, the emergence of ecosystems represented a shift from isolated forms of life to interconnected networks. It was a transition from individual survival to collective harmony, where organisms influenced not only their own fates, but the fates of others. The ocean was no longer just a backdrop for life. It became a dynamic environment shaped by the interactions of its inhabitants. Each organism, no matter how small, contributed to the balance of the whole. These early ecosystems set the stage for all future environments, from coral reefs to forests to grasslands, each built on the same principles of interdependence and gentle exchange. The formation of ecosystems reminds us that life is not merely a collection of individuals, but a tapestry woven from countless interactions. In the ancient oceans, where tissues, organs, and systems were beginning to take shape, ecosystems added another layer of organization, one that extended beyond the boundaries of single organisms and embraced the unity of life on a planetary scale. 14. The power of evolutionary variation. Life learns to diversify. In the continuing calm of Earth's ancient oceans, long after tissues, organs, and early ecosystems began to shape the living world, another quiet but foundational process steadily guided life's unfolding story. This process, subtle but persistent, was variation, the gentle differences that arose from one generation to the next. Variation did not arrive with fanfare or sudden leaps. It emerged naturally from the way living things replicated, responded to their surroundings, and interacted within their environments. Yet, through countless cycles of reproduction, these small differences accumulated, giving life the flexibility and resilience it needed to survive changing conditions. Variation was the soft current beneath life's expansion, carrying organisms into new forms, new abilities, and new realms of possibility. To understand variation in its earliest forms, imagine simple organisms drifting through the slow-moving currents of the primordial seas, each cell divided at its own pace, creating offspring nearly identical to itself. These tiny imperfections, slight changes in structure, minor shifts in metabolic efficiency, subtle adjustments in sensitivity to light or temperature, became the seeds of variation. No organism intended to change. Variation occurred simply because the processes of life, delicate as they were, left room for small differences. Some of these differences had no immediate effect. Others provided subtle advantages, allowing certain organisms to endure environmental shifts a little better than their neighbors. As early ecosystems grew more complex, the environment became a soft but constant filter through which variation passed. Organisms that were slightly better suited to their surroundings tended to survive longer, reproduce more often, and contribute their traits to future generations. This gentle filtering was not directed or planned, it was the natural result of differences in survival and reproduction. In this way, variation became the foundation of adaptation. Over long periods, populations of organisms slowly shifted their characteristics, becoming better matched to the conditions of their habitats. Some grew accustomed to brighter sunlight near the surface. Others adapted to the dim, colder depths. Variation allowed life to test many possibilities simultaneously, exploring countless pathways toward greater stability. The emergence of eukaryotic cells, with their nuclei and organized genetic systems, enhanced the potential for variation. Their genetic material was protected, yet also capable of more intricate patterns of replication and mutation. When these cells divided, their chromosomes occasionally changed in subtle ways, generating new combinations of traits. These genetic variations built upon the simpler molecular changes that had guided earlier evolution. Organisms now had more ways to explore the landscape of biological possibility, developing not only new structures but new behaviors and interactions. Variation extended beyond individuals to entire populations. As organisms spread into different regions of the ocean, they encountered distinct conditions, variations in temperature, light, salinity, and nutrient availability. These subtle differences influenced which traits were most successful in each environment. Over time, populations diverged. Organisms in warmer waters developed different characteristics from those in cooler depths. Those living near hydrothermal vents adapted to minerals and heat, while those near the surface adapted to sunlight and oxygen. These gradual divergences laid the foundation for the earliest forms of speciation. Though the boundaries between species were not yet sharp, life was exploring different paths, shaping itself in quiet conversation with the environment. Variation also influenced the interactions within ecosystems. Predators developed traits that helped them capture prey more efficiently, while prey developed traits that helped them avoid detection or escape. Producers adapted to maximize their absorption of sunlight or nutrients, while decomposers became more efficient at breaking down complex molecules. These interactions created a dynamic equilibrium, with each group influencing the evolution of the others. Through these gentle pressures, life's diversity deepened. Variation ensured that no ecosystem remained static. It transformed simple communities into rich networks of interdependence and subtle change. As multicellularity developed, variation gained even more complexity. Cells within organisms began specializing in ways that produced differences not within populations but within individuals. These differences allowed tissues and organs to perform distinct tasks, adding layers of internal variation that contributed to the organism's stability. Even at this early stage, internal variation hinted at the intricate biological patterns that would later allow for nervous systems, circulatory networks, and coordinated movement. These innovations did not arise suddenly but accumulated as life explored different combinations of structures and functions, guided by the quiet influence of variation. Environmental changes also played a role in shaping variation. Earth's climate shifted over long cycles of warming and cooling, influencing ocean currents, nutrient distributions, and available habitats. Volcanic activity altered atmospheric composition, while continental drift gradually reshaped the ocean floor. Life encountered these shifts not with resistance, but with adaptability. Variation allowed organisms to adjust, finding new ways to survive as the world changed around them. Even dramatic events, such as periods of increased oxygen or temporary drops in temperature, were softened by the resilience variation offered. It gave life the capacity to flow with the planet's rhythms rather than break against them. Through variation, life also learned to experiment. Some organisms developed unusual shapes or metabolic pathways that, while not immediately advantageous, opened the door to future possibilities. These exploratory variations allowed life to innovate gently, trying new configurations without abandoning older ones. In this way, variation served as both a stabilizing force and a creative one. It ensured continuity but also allowed for novelty, weaving both threads into the tapestry of evolution. As populations continued to diverge, ecological roles became more defined. Some organisms specialized in capturing sunlight, others in grazing on producers, still others in scavenging or decomposing. Variation allowed organisms to spread into every available niche, filling the oceans with a mosaic of living forms. This diversification strengthened ecosystems by creating redundancy and balance. If one group declined, others often filled similar roles, maintaining the gentle flow of energy and nutrients. The diversity born of variation thus helped ecosystems remain stable even as conditions shifted. Looking back on this period, it becomes clear that variation was the quiet engine of evolution. It worked softly, without force, guiding the gradual transformation of life through countless small changes. It shaped organisms from within and woven their relationships across entire ecosystems. Through variation, life developed resilience, flexibility, and diversity, the qualities that would one day allow it to leave the oceans, explore the land, and fill the planet with forms and colors beyond imagination. In the gentle currents of ancient Earth, variation reminded life that change was not something to fear but something to embrace. It offered opportunity, stability, and creativity, and through its patient influence, it laid the foundation for the increasingly intricate forms that would soon begin to emerge. 15. Natural selection as a quiet sculptor. In the long, gentle story of life's unfolding, after variation began to spread through early populations and ecosystems gained depth and resilience, another quiet force emerged as a steady guide. This force did not create new traits itself, nor did it push life in any predetermined direction. Instead, it acted softly, shaping the possibilities that variation produced. This was natural selection, a process so subtle in each moment that its effects could only be seen over vast stretches of time. And yet, through its patient influence, natural selection became one of the most important sculptors of life's diversity, refining forms, strengthening adaptations, and guiding organisms toward ways of living that allowed them to flourish. To imagine natural selection in its earliest forms, picture the ancient oceans filled with countless organisms, each harboring slight differences inherited from their ancestors. Some differences were matters of chance. A cell divided slightly faster, a pigment absorbed sunlight more efficiently, a membrane proved marginally more stable in warmer water. These traits were not designed or intended. They simply existed, emerging from the gentle imperfections in replication and the subtle influences of the environment. Yet each trait carried with it a quiet potential, an opportunity to persist or to fade away. Natural selection entered this landscape not as a force pushing life forward, but as a soft filter. When conditions changed, when light dimmed, when nutrients shifted, or when predators became more common, some organisms were slightly better suited to the new reality. They survived just a bit longer, reproduced a bit more often, and passed their traits to the next generation. Over countless cycles of birth and death, these small advantages accumulated. The ocean did not notice, The organisms themselves did not understand, but slowly, gently, populations changed. Traits that improved survival became more common, while those that hindered survival became rare. Natural selection also worked through interactions between organisms. In ecosystems where predators grazed upon smaller creatures, any variation that helped prey avoid detection or escape provided an advantage. A slightly faster movement, a faint shift in coloration, or a tendency to gather with others could all make survival more likely. Over time, these traits were gently favored. Predators, in turn, responded through their own variations. Some developed sharper senses, others refined their methods of movement, and still others improved their ability to digest particular types of prey. This give and take was not a battle, but a series of subtle adjustments, each shaping the other in a continuous, interwoven dance. Natural selection also guided the emergence of cooperation. In early multicellular organisms, cells with traits that supported their neighbors created stronger, more resilient structures. A cell that produced a slightly more durable form of connective material helped the entire organism maintain stability. A cell that responded gently to chemical signals contributed to coordinated movement or shared growth patterns. These traits, beneficial to the group, became more common. Over long stretches of time, these tendencies laid the foundation for tissues, organs, and complex systems, structures that could only exist when cells behaved in concert, guided by the quiet influence of selection. Environmental changes played a central role in this sculpting process. The rise of oxygen in the atmosphere created new challenges and opportunities. Organisms with traits that allowed them to tolerate or use oxygen thrived, while those without such traits retreated into oxygen-poor environments. Shifts in temperature created new ecological niches. Currents moved nutrients across vast distances, creating pockets of abundance and scarcity. Natural selection responded to these shifts gently but consistently, encouraging traits that supported survival and reproduction in each new landscape. Even the emergence of early organs and organ systems was shaped by selection. Structures that improved nutrient absorption, protected delicate cells, or supported efficient movement provided small but meaningful benefits. An organism with a slightly more effective digestive cavity could extract more energy from its surroundings. Another, with a thicker protective layer, could withstand physical stress more easily. Over time, these advantages accumulated. Though each step was subtle, the cumulative effect was profound, guiding multicellular life toward greater sophistication. Natural selection also encouraged balance within ecosystems. When populations grew too large for their food sources, competition increased. Variations that allowed some individuals to exploit new resources or survive on less helped stabilize the population. When predators became too abundant, their own food sources declined, reducing predator numbers and allowing prey populations to recover. Through these gentle cycles, selection helped maintain a dynamic equilibrium, ensuring that no single group overwhelmed the others. This balance supported the long-term continuity of ecosystems, allowing them to persist through gradual changes. An essential aspect of natural selection was its relationship to heredity. Traits that aided survival were only meaningful if they could be passed to future generations. In early life, genetic material became increasingly stable and reliable, supported by the protective nature of the nucleus in eukaryotic cells. This stability allowed adaptive traits to persist, creating a lineage of organisms shaped by their ancestors' successes. Through reproduction, life learned to carry its past forward, refining itself gently through each generation. Natural selection also laid the groundwork for complexity by encouraging the emergence of traits that supported multicellularity. Organisms that could coordinate their internal processes more effectively, communicate between cells, or regulate growth patterns gained subtle advantages. Over millions of years, these advantages shaped organisms that were more structured, more resilient, and more capable of exploring new environments. Natural selection did not rush this process. Instead, it worked patiently, allowing complexity to arise naturally through the accumulation of beneficial traits. Through all these changes, natural selection remained consistent in its quiet influence. It did not eliminate variation but shaped it. It did not predict the future but responded to the present. It did not act with intention but through the simple reality that organisms better suited to their environments left more descendants, and through this gentle, unassuming process, life grew in diversity, form, and function. As we reflect on this chapter in life's long narrative, it becomes clear that natural selection served not as a force of struggle but as a guide toward harmony, helping organisms adapt to their surroundings, find balance within ecosystems, and build structures that would support the journey toward even greater complexity. It was the quiet sculptor, shaping life not with heavy strokes, but with countless subtle touches, each one contributing to the beauty of the living world. 16. The rise of adaptation. Life responds to its changing world. In the gentle continuity of Earth's ancient story, after natural selection began guiding populations through subtle shifts and ecosystems found their first rhythms of balance, another quiet movement took shape, one that would allow life not merely to persist, but to flourish. This movement was adaptation, the gradual, soft, and deeply patient process in which organisms adjusted themselves to the environments they inhabited. Adaptation was not a sudden transformation, nor was it a dramatic leap. Instead, it was a quiet unfolding, a slow refinement of traits shaped by the interplay between variation and natural selection. Through adaptation, life learned to settle more comfortably into the contours of its surroundings, responding to light, temperature, pressure, nutrients, and the presence of other organisms with increasing elegance and precision. To imagine adaptation in its earliest forms, picture the ancient oceans, where diverse organisms drifted, swam, or anchored themselves in calm sediment. Each organism carried a unique set of traits, subtle differences, shaped by countless generations of variation. Some absorbed light more efficiently, others could withstand colder waters, and still others were better suited to capturing nutrients in low-resource environments. These traits did not arise because organisms anticipated change, but because their ancestors had experienced slight differences in survival and reproduction. Over time, traits that supported survival became more common, gently guiding populations toward forms better suited to their surroundings. Adaptation was a dynamic process, responding to conditions that shifted slowly across geological time. As oxygen levels in the atmosphere increased, organisms that could tolerate or use oxygen gained a quiet advantage, expanding into environments previously inaccessible. Others retreated into oxygen-poor niches, adapting to survive in the darker, deeper layers of the ocean. Adaptation allowed life to sort itself into these new spaces with remarkable grace. Oxygen, once toxic to early organisms, gradually became a source of energy for more advanced cells. This transformation, rooted in adaptation, opened pathways toward greater metabolic efficiency and laid the foundation for future complexity. Environmental gradients also encouraged adaptation. Near the ocean's surface, sunlight varied in intensity depending on depth, time of day, and clarity of water. Organisms adapted to these subtle variations by developing pigments suited to specific wavelengths of light. Those living closer to the surface benefited from pigments that protected them from ultraviolet radiation while still capturing energy efficiently. Deeper-dwelling organisms evolved pigments optimized for the soft, filtered light that reached them. These adaptations allowed life to spread across vertical layers of the ocean, creating ecological diversity that supported more intricate food webs. Adaptation also played a central role in shaping the movement and behavior of early organisms. Some species developed flagella, gentle, whip-like structures that allowed them to move slowly through the water. This movement enabled them to reach nutrient-rich areas or escape regions that grew too warm or too crowded. Other organisms adapted by becoming more streamlined, reducing energy expenditure as they drifted with currents. Still, others developed the ability to sense chemical gradients, allowing them to navigate towards sources of nutrients or away from potential danger. These sensory and motility adaptations strengthened survival and enriched interactions within ancient ecosystems. In multicellular organisms, adaptation influenced the development of tissues and organs in new and subtle ways. Tissues exposed to different environmental pressures began to specialize more deeply. Organisms living in brighter regions expanded their photosynthetic tissues, increasing surface area to capture more light. Others living in environments with scarce nutrients developed thicker storage tissues or more efficient internal transport systems. These adaptations refined the structure and function of early multicellular bodies, helping them maintain stability and respond gently to the dynamic world around them. Predator and prey also adapted in gentle tandem. Early predators, even in their simplest forms, gained advantages when they developed traits that made capturing other organisms more efficient. A slight increase in speed, a more sensitive detection pathway, or a more effective feeding structure allowed them to secure more energy. Prey responded with their own adaptations. Some became more transparent to blend into the water, making detection more difficult. Others developed stronger outer layers for protection or reproduced more rapidly to sustain their populations despite predation. Over time, this continuous dance of adaptation helped create balance within ecosystems, ensuring that no single group overwhelmed the others. Adaptation extended to the internal regulation of organisms as well. Early life needed to maintain stable internal environments despite shifting external conditions. Cells adapted by developing more refined membranes, capable of regulating the flow of ions and molecules. These membranes protected delicate internal processes, allowing cells to function reliably even when the surrounding water changed in salinity or temperature. As multicellular organisms grew, internal adaptation became even more important. Systems for distributing nutrients and removing waste became more efficient, ensuring that cells deep within the organism received what they needed. These internal adaptations supported increasing size and complexity, providing the groundwork for future innovations. Geological changes also shaped life through adaptation. Shifting continents, volcanic activity, and changes in sea level created new environments and removed others. Organisms that once thrived in warm, shallow seas found themselves adapting to deeper or cooler waters. Those adapted to coastal regions migrated or developed strategies for surviving periodic drying or exposure. Adaptation allowed life to follow the changing shape of the planet, turning potential challenges into opportunities for diversification. One of the most profound aspects of adaptation was its cumulative nature. Each small shift in physiology, behavior, or structure built upon earlier changes, creating a gentle layering of innovations across deep time. A pigment adapted for absorbing certain wavelengths might later support the development of more complex photosynthetic systems. A basic sensory mechanism might evolve into intricate communication pathways. A structural tissue adapted for support might eventually give rise to skeletal frameworks. Adaptation did not merely solve immediate challenges, it created pathways along which future evolution could flow. As adaptation continued to sculpt organisms, the diversity of life deepened. Populations became more distinct as they adjusted to different conditions. Ecosystems grew more intricate as each species developed traits suited to its role. The world became a tapestry of forms and behaviors, each woven through countless generations of quiet change. From microbes drifting near hydrothermal vents to simple multicellular organisms anchoring themselves on ancient shorelines. In reflecting on this chapter of life's story, one can see adaptation not as a struggle, but as a gentle alignment, a gradual tuning of organisms to the environments that supported them. It was a calm, patient response to change, guided by variation and shaped by natural selection. Through adaptation, life discovered resilience, versatility, and the ability to explore every opportunity the Earth offered. It was through countless small adaptations that life prepared itself for the next great transitions, which would introduce even more complexity, diversity, and interdependence. 17. Emergent properties and the magic of integration. As life continued its gentle expansion across the ancient seas, shaped by variation, guided by natural selection, and refined through gradually unfolding adaptations, an even deeper layer of complexity began to reveal itself. This layer was not tied to any single structure, molecule, or cell. Instead, it arose from the way parts interacted, how individual components, when linked together in quiet cooperation, produced new qualities that none of them possessed alone. These were emergent properties, the subtle and beautiful outcomes of integration. Their appearance marked a turning point in the story of life, showing that complexity does not arise merely from accumulating new parts, but from the harmonious relationships that grow between them. To understand emergence in its earliest forms, imagine again the simple multicellular organisms drifting calmly through ancient oceans. At first glance, they appear as modest groups of cells, each performing familiar tasks, absorbing nutrients, responding to chemical signals, or dividing when necessary. But when these cells began interacting in coordinated ways, something entirely new appeared: the ability to behave as a unified whole. No single cell carried the instructions for this unity. Instead, it emerged from the collective, from the ways cells exchanged signals, shared resources, and responded to their neighbors. This phenomenon, where cooperation created capabilities far beyond the reach of isolated cells, was one of the earliest examples of emergent properties shaping life's gentle evolution. Emergence also played a significant role inside individual cells. Early eukaryotic cells contained organelles, mitochondria, nuclei, ribosomes, each performing its specialized task. Yet the cell itself possessed qualities that none of these organelles could exhibit on their own. A mitochondrion could not sense or react to the environment alone. A nucleus could not maintain homeostasis outside the protective interior of the cell. A ribosome could not build a functioning protein without the guidance of genetic material and the presence of other molecules. But when all these components worked together within a shared boundary, they generated the intricate choreography that made cellular life possible. The cell could sense, respond, repair, and regulate. These capacities did not belong to any part in isolation. They emerged softly from the integration of all parts working in harmony. As multicellularity advanced, emergent properties became even more pronounced. A single cell could not anchor an organism to the seabed, but a layer of cells working together formed tissues that provided structural stability. A single photosynthetic cell could absorb sunlight, but a sheet of interconnected cells could capture far more energy, supporting larger bodies and more complex structures. Similarly, digestive tissues transformed from simple sacs into coordinated systems, capable of moving nutrients, breaking them down, and distributing them internally. None of these functions could have emerged without the interplay between tissues, molecules, and structures acting in unity. Emergence is often described as the whole being greater than the sum of its parts. But in the peaceful unfolding of early life, it may be better understood as a gentle rising new qualities floating upward from cooperation. These new properties were not imposed from above. They appeared naturally whenever systems reached a certain level of complexity. The early ocean was full of such transitions. When molecules formed membranes, they gained the ability to trap chemical gradients. When vesicles grew larger, they gained the ability to divide. When simple networks of reactions became more stable, they developed the basic qualities of metabolism. At each stage, emergence guided life to new levels of intricacy without the need for sudden leaps. One of the most profound emergent properties was communication. Cells produced chemical signals that shaped the behavior of their neighbors. These signals allowed entire tissues to contract rhythmically or expand gently in response to environmental cues. Over time, Communication networks grew more refined, allowing organisms to respond not just reflexively, but in coordinated, balanced ways. Such responsiveness represented a form of intelligence, not conscious thought, but a distributed awareness arising from countless tiny interactions. This emergent intelligence helped organisms navigate their surroundings, maintain internal stability, and adapt gracefully to changing conditions. Emergent properties also influenced reproductive strategies. Simple organisms that produced spores or reproductive cells discovered that coordinated timing improved success. Populations that released reproductive material together increased their chances of forming new colonies. This synchrony was not planned. It emerged from the tendency of organisms to respond similarly to shared environmental cues such as light, temperature, or the availability of nutrients. Over time, these coordinated reproductive strategies strengthened populations and encouraged further diversification. In ecological settings, emergence shaped the interactions between organisms and their environments. When producers, consumers, and decomposers interacted, ecosystems formed behaviors that none of their individual species possessed. Nutrient cycles, energy flows, and ecological stability arose from the collective activities of countless organisms. An ecosystem could recover from disturbances, regulate its internal composition, or maintain balance across seasons, not because any single organism orchestrated these changes, but because the system as a whole responded dynamically to shifting conditions. In this way, ecosystems themselves exhibited emergent properties, becoming self-regulating and resilient through the cooperation of their inhabitants. Even the evolutionary process exhibited emergent qualities. Natural selection acted on individual variations, yet over long periods, entire lineages transformed in gentle, sweeping arcs. New species emerged, ecosystems diversified, and global patterns of life shifted. This overarching movement was not directed by any external force. It emerged from the interactions between organisms, their environments, and the persistent flow of time. Evolution was not merely the sum of individual adaptations, it was the unfolding of complexity arising from countless, interwoven processes. Inside organisms, emergent properties prepared life for future complexity. The development of coordinated tissues allowed for organs. Organ systems interacting gave rise to physiological regulation. Communication networks within organisms, once simple chemical signals, gradually laid the foundation for more sophisticated coordination, eventually leading to nervous systems and, later still, to consciousness itself. These later developments remained far in the future during the early ages described here, yet their roots were already present in the quiet interactions of ancient cells. What makes emergent properties especially calming to imagine is their effortless nature. They arise not through sudden invention, but through connection through the patient accumulation of relationships. Life did not need to design new traits from scratch. It only needed to interact, cooperate, and allow complexity to flow naturally from those interactions. Emergence reveals that life's richness comes not only from its individual parts, but from the ways those parts come together in soft, harmonious union. As we reflect on this period of biological history, it becomes clear that emergence was the gentle force that lifted life from simplicity into ever-deepening complexity. It allowed molecules to become cells, cells to become organisms, and organisms to weave themselves into ecosystems. It demonstrated that new qualities, stability, coordination, awareness, resilience, could arise from collaboration rather than from competition alone. This quiet magic of integration prepared the world for the next steps in life's journey, steps that would continue to build upon the subtle foundations laid in the ancient seas. Through emergence, life learned that the most extraordinary transformations often come not from abrupt change, but from the patient strengthening of connections. 18. The Cambrian prelude: quiet foundations before the burst. In the tranquil sweep of Earth's deep past, long before vibrant animals swam through illuminated seas, or complex creatures walked upon the land. There was a period in which life gathered itself with calm deliberation. It was a time of preparation, when the foundations for future diversity were laid softly, layer by layer. Scientists often speak of the Cambrian explosion, the moment when animal life blossomed dramatically into recognizable forms, but this extraordinary flowering did not arise suddenly from silence. Instead, it emerged from a long, gentle prelude in which early organisms refined their bodies, their tissues, and their interactions with the world. This chapter, the Cambrian Prelude, reveals how life quietly built the scaffolding upon which complexity would later rise. To picture this era, imagine the ancient oceans still dominated by simple multicellular organisms. They floated, anchored, or drifted in soft assemblies, forming colonies, mats, or tube-like structures. Though these organisms lacked the intricate organs of later animals, they had begun to develop increasingly coordinated tissues. Some had internal cavities that circulated nutrients more effectively. Others formed external layers that behaved like early skins, protecting delicate internal cells from the ocean's shifting chemistry. These gentle improvements strengthened life's resilience and prepared it for the changes to come. One of the most important developments of this prelude was the quiet refinement of symmetry. Early organisms experimented with shapes that allowed more efficient interaction with their surroundings. Some adopted radial symmetry, spreading their structures evenly around a central axis. This shape allowed them to sense and absorb nutrients from all directions, a useful strategy for creatures that lived anchored to one place. Others began to explore bilateral symmetry, where the body developed mirrored left and right sides. This simple form of organization would later become essential for movement, coordination, and the development of directed behaviors. At this early stage, however, symmetry was still emerging softly, arising through subtle shifts in growth patterns and cellular arrangement. During this gentle period, early animals known collectively as ediacaran organisms began to populate the ocean floors. Their forms were soft, delicate, and often strangely beautiful. Some resembled fronds swaying in the water, with rippling surfaces designed to increase nutrient absorption. Others were disc-like or quilted, lying flat on the seafloor where they exchanged gases and nutrients with their surroundings. These organisms did not yet possess true muscles, mouths, or digestive tracts, but they represented an important step toward more complex body plans. Their existence showed that life had begun to explore multicellular organization beyond simple colonies, moving toward integrated forms that could gently coordinate their internal and external functions. The ocean environment of this time also encouraged new patterns of interaction. As oxygen levels continued their slow rise, organisms gained access to more efficient metabolic pathways. This improvement allowed them to grow larger and maintain more active lifestyles. Still, the world remained calm compared to the vibrant ecosystems that would follow. Predation was minimal, with only the earliest hints of organisms feeding on one another. Most life continued to rely on passive absorption or gentle filtering of nutrients. In this peaceful setting, organisms had the freedom to explore structural and developmental innovations without intense ecological pressure. One of the key innovations emerging quietly in this prelude was the refinement of developmental processes. Early animals began experimenting with layers of tissues that formed during embryonic development. From these layers, ectoderm, mesoderm, and endoderm future organs and systems would one day arise. Even though these early organisms remained simple, the beginnings of developmental organization were already taking shape. Cells were learning when to divide, when to differentiate, and how to form structures in predictable arrangements. This coordination, though primitive, marked a profound shift. Life was beginning to plan itself from within, following gentle patterns that would eventually guide the formation of muscles, nerves, and digestive systems. During this time, the oceans also saw the rise of soft-bodied organisms capable of movement. Though slow and simple, these movements represented a new form of agency. Some organisms glided across microbial mats, leaving faint impressions in the sediment. Others performed rippling or contracting motions that allowed them to reposition themselves slightly in response to light, currents, or nutrients. These movements, though minimal, required tissues and cellular networks refined enough to coordinate action across the organism. They were early steps toward locomotion, a capacity that would later transform ecosystems. The Cambrian prelude also witnessed the gradual emergence of ecological layering. Organisms began occupying different depths, substrates, and environmental niches. Some settled on the seafloor, forming carpets of life that filtered nutrients from the water. Others floated freely, drifting gently with currents. Still others anchored themselves to rocks, vents, or sediments. This diversification of habitats created opportunities for new interactions and increased the potential for adaptive differentiation. The ocean, once dominated by simple forms, began to resemble a more complex mosaic, with life spreading into spaces made possible by the gradual refinement of bodily structures. One of the most enchanting aspects of this era is how life prepared for future leaps without knowing what would come. Muscles had not yet fully formed, yet tissues began developing the contractile proteins that would later support movement. Nervous systems had not yet appeared, yet communication pathways became increasingly refined, allowing cells to respond more effectively to one another. Hard skeletons did not yet exist, yet some organisms began strengthening their outer layers with proteins and minerals. The foundations of eyes, mouths, limbs, and shells were not yet present, but their precursors, the subtle changes in tissue arrangement and developmental chemistry, were quietly unfolding beneath the surface. In many ways, the Cambrian prelude was a time of potential gathering itself. Like the soft swelling of a tide before it rises, life's internal organization was becoming more flexible. Body plans were diversifying. Ecosystems were becoming richer and more layered. Environmental conditions were stabilizing in ways that supported future innovation. The Earth itself seemed to be preparing for a moment when complexity could blossom into forms never seen before. Yet despite this preparation, the world remained peaceful. The oceans still held slow, drifting organisms. Soft-bodied creatures continued their gentle existence upon the seafloor. The rhythms of life were unhurried, guided by currents, sunlight, and the quiet chemistry of ancient waters. There was no urgency, only the patient unfolding of possibility. When the Cambrian explosion would eventually arrive, bringing animals with hard parts, complex sensory systems, and active predation, it would represent the culmination of these soft beginnings. But for now, the planet rested in a state of quiet readiness, shaped by the subtle emergence of traits that would one day define the biological world. 19. The quiet dawn of complexity, the world before the bloom. In the soft rhythm of Earth's ancient ages, as the oceans continued their slow transformations and early ecosystems deepened in their interconnectedness, life was quietly preparing for one of its most remarkable transitions. This was the threshold just before true complexity blossomed, a period often overshadowed by the dramatic burst of the Cambrian explosion that would follow. Yet this preceding era, gentle and contemplative in its pace, carried within it the seeds of everything that would soon emerge. It was a time when simple organisms refined their internal patterns, when developmental pathways grew more stable, and when the subtle foundations of complex body plans took form. This quiet dawn of complexity was not a moment of spectacle, but a slow accumulation of potential, a gathering of threads that would later weave the vibrant tapestry of animal life. To envision this time, imagine a world filled with tranquil seas, their surfaces shimmering beneath soft, ancient sunlight. The organisms inhabiting these waters were still mostly simple, soft-bodied beings, but they had become far more organized than their earliest predecessors. Many of these creatures belonged to the Ediacaran biota, a diverse array of forms that drifted, crawled, or lay gently upon the seafloor. Some appeared as fronds, standing upright like delicate underwater leaves. Others formed wide discs or elongated ribbons, resting in quiet symmetry along the sediment. Though lacking the complex organs and musculature that later animals would possess, these creatures displayed coordinated tissues and repeating patterns that hinted at deeper biological structure. In these early forms, the faint outlines of more complex developmental plans began to appear. Certain organisms organized their tissues into layers that resembled the beginnings of germ layers, the foundational tissues from which future organs would arise. Cells learned, in simple but significant ways, how to follow developmental cues that guided their differentiation. These early patterns did not yet produce eyes, limbs, or circulatory systems, but they provided the underlying structure from which such features could eventually evolve. The body plans of the future were already present in essence, like soft sketches waiting to be refined. One of the most remarkable features of this pre-Cambrian world was the emergence of larger body sizes. With more efficient nutrient distribution, improved cellular cooperation, and rising levels of oxygen in the water, organisms could grow beyond the tiny forms that dominated earlier ages. Larger size allowed new kinds of interaction with the environment. It altered how organisms captured nutrients, how they absorbed oxygen, and how they responded to predators, even though predation was still in its infancy. Growth also encouraged new shapes. Some organisms branched repeatedly, forming fractal-like structures that maximize surface area. Others developed broad, flat bodies suited to capturing light or filtering nutrients from the water. As the oceans filled with these gently expanding forms, ecosystems grew richer in subtle but meaningful ways. The seafloor, once a relatively sparse landscape, became a soft mosaic of organisms layered, intertwined, and dependent on one another's presence. Some species formed dense colonies that created stable habitats. Others grazed softly upon microbial mats, shaping the distribution of nutrients. Even the simple movement of water stirred by larger organisms contributed to the mixing of sediments and the flow of resources. Though the interactions remained peaceful and slow compared to what would follow, they marked the beginning of more dynamic ecological relationships. During this period, the first signs of movement began to appear with greater clarity. While many organisms still relied on passive drifting, some developed tissues capable of gentle contraction, allowing them to glide or shift themselves slightly across soft substrates. These movements were not yet intentional in the way we think of animal motion, but they represented the early emergence of locomotion. A creature that could move even slightly had access to more varied environments, regions richer in nutrients, spaces with different light levels, or areas less crowded by other organisms. Movement also introduced the first hints of animal-environment feedback, as organisms altered the sediments they traveled across, creating patterns that would later fossilize into quiet signatures of ancient life. At the same time, the earliest forms of predation began to arise. Though still extremely limited, this shift had profound implications. A few organisms developed structures or behaviors that allowed them to consume other multicellular beings rather than relying solely on dissolved nutrients. These early predators were not the swift, active hunters of later ages. Instead, they likely relied on slow, engulfing movements or simple digestive mechanisms that could break down soft tissues. Yet even this subtle introduction of predation placed new pressures on ecosystems. Prey organisms began to adapt with slightly tougher tissues, broader shapes that made engulfment more difficult, or growth patterns that lifted part of the body above the substrate. In this gentle dance between predator and prey, selection pressures deepened, preparing the world for the rapid diversification that would soon occur. Another quiet but essential development of this era was the stabilization of genetic and developmental mechanisms. Genetic regulatory networks, systems that control when and how genes are activated, grew more coordinated. These networks allowed organisms to develop consistent body patterns, generation after generation, creating the stability needed for more complex traits to evolve. Some scientists refer to these networks as the genetic toolkit, a set of regulatory systems that would later give rise to limbs, shells, segmentation, and complex sensory structures. In the peaceful prelude to the Cambrian, this toolkit was being assembled piece by piece, though it had not yet been used to its full potential. Environmental conditions also played a quiet but powerful role. Continental configurations influenced nutrient flow, while shifts in climate affected ocean chemistry. Increased oxygen levels allowed for more energetically demanding lifestyles. Mineral availability changed, laying the groundwork for the later evolution of hard parts like shells and exoskeletons. Even the ocean floor itself was transforming, shaped by microbial activity that created textured mats. These mats provided substrates for organisms to rest upon or burrow through, encouraging further ecological experimentation. As all these elements came together, body plans stabilizing, tissues refining, oxygen rising, ecological interactions deepening, and genetic toolkits assembling, the stage was set for something extraordinary. But the extraordinary had not yet arrived. Instead, the world remained in a state of gentle preparation, filled with soft-bodied creatures living quiet lives beneath ancient waters. They were the architects of possibility, laying the foundation for forms that would not appear until long after their own time. Their existence reminds us that complexity does not always begin with sudden change. Sometimes it begins with a stillness that gathers strength over ages. The Cambrian explosion, when it arrived, would fill the oceans with shells, eyes, limbs, and intricate body structures. But without this peaceful era, the quiet dawn preceding the bloom, such rapid diversification could never have occurred. The world needed this time. Life needed this time. In the whispering currents of the Ediacaran seas, evolution was preparing Earth for one of its most creative chapters. 20. The long journey toward consciousness, the earliest sparks of awareness. In the soft, unhurried flow of Earth's ancient past, after life had refined its structures, deepened its ecosystems, and prepared itself for the dramatic bloom of diversity to come, a final thread began to weave quietly through the tapestry of biological complexity. This thread was awareness, not in the elaborate, reflective form that humans experience today, but in the simplest and earliest sense of the word. It was the emergence of responsiveness, the gentle awakening of organisms to the subtle cues of their surroundings. This early awareness was not thought, nor intention, nor anything resembling consciousness as we know it. Instead, it was the faint spark of sensitivity that allowed life to engage with the world in new and transformative ways. It was the beginning of perception, coordination, and the first stirrings of behavior shaped from within. To imagine these earliest sparks, picture the oceans just before the Cambrian bloom. Soft-bodied organisms moved slowly across the sea floor, their tissues increasingly capable of sensing differences in light, temperature, pressure, and chemical gradients. They could not see or hear, but they could register changes. A slightly brighter patch of water signaled shallow sunlight above. A shift in chemical concentration hinted at food drifting nearby. A gentle pressure on the body revealed a passing current or the approach of another organism. These sensations were faint and diffuse, but they represented an important step. Life was beginning to form internal maps of its environment, however primitive, and to respond accordingly. At first, the responses were simple. A slight contraction when touched, a gentle movement toward or away from a stimulus, a shift in growth direction when nutrients became scarce. These behaviors required no centralized control, no brain, and no organized nervous system. They were emergent outcomes of communication within tissues, chemical signals passing from cell to cell, coordinating action in a way that mimicked the earliest instincts. Even in these simple forms, life was beginning to display patterns of behavior shaped by the environment and refined by natural selection. The gradual increase in oxygen played a quiet role in this awakening. As oxygen became more available, cells could support more energy-demanding processes. This allowed tissues to develop proteins that responded faster to stimuli, channels that regulated internal chemistry more precisely, and pathways that transmitted signals across greater distances. Internal coordination grew more refined. Responses became more specific. With time, organisms gained the ability not just to react, but to adjust their behavior based on repeated experiences, choosing, in the simplest biological way, what benefited them most. These early sensory and behavioral capacities laid the groundwork for the first nervous systems. Long before neurons existed, cells began forming networks that transmitted signals more efficiently than passive diffusion could allow. Some cells specialized in sending rapid chemical or electrical impulses. Others specialized in receiving them. These subtle differences marked the quiet beginnings of excitability, a property that would later allow nerves to transmit information with remarkable speed. But at this early stage, the networks were still diffuse and simple, barely distinguishable from ordinary tissues, yet harboring the seeds of astonishing future complexity. In these calm and ancient seas, the first faint division between self and environment began to appear. Organisms could distinguish between their internal balance and external conditions. They developed the capacity to maintain homeostasis, to protect the harmony within their cells even as the world around them shifted. This inner stability, paired with subtle external responsiveness, formed the earliest bridge between biology and awareness. It marked the moment when life began not only to exist, but to experience. Behavioral emergence continued gently across generations. Some organisms responded to light by drifting upward, seeking brightness that supported photosynthesis. Others responded by drifting downward, avoiding exposure or predators that had begun to appear. Over time, organisms that could respond more precisely to environmental cues survived more successfully. Natural selection favored the refinement of sensory pathways, leading to increasingly sophisticated networks of cells that transmitted information across the body. As organisms grew larger and more structurally diverse, coordination became even more important. Different tissues needed to act in harmony, contracting, expanding, or shifting in controlled ways. Early sensory cells evolved in tandem with early motor cells, forming the first rudimentary sensory motor loops. These loops allowed organisms to detect changes and respond in a unified manner. Though still vastly simpler than later organisms, these early systems represented a major milestone in the story of life. They laid the earliest framework for movement, guided by sensation, a principle that would shape animal evolution profoundly. In ecological contexts, these developments deepened the complexity of interactions. Predators became slightly more adept at detecting prey. Prey became more adept at sensing danger. Filter feeders grew responsive to currents that carried nutrients. Sessile organisms adapted their growth patterns based on light and chemical gradients. Awareness, even in its early, unfocused form, helped shape ecological balance. It allowed organisms to find more favorable habitats, avoid challenges, and explore new niches. Through these subtle influences, awareness contributed to the growing diversity that characterized the Precambrian world. One of the beautiful aspects of this early awakening is its humility. There was no sudden leap into consciousness, no dramatic shift. Instead, awareness grew slowly, building itself through countless interactions. A chemical receptor sensing a nutrient, a cell membrane shifting in response to pressure, a colony of tissues contracting in coordinated rhythm. Each of these small events represented the unfolding of sensitivity and the beginnings of perception. Together, they shaped the biological landscapes that would later support nervous systems, brains, memory, and the rich inner worlds of animals. As time drifted forward, these early behaviors and sensitivities created the conditions necessary for the Cambrian explosion. Organisms were now able to move with more coordination. They could sense more clearly. They could interact more deeply with one another. The world was no longer a place of passive drifting and gentle filtering. It was becoming a stage on which life would soon explore more active, dynamic roles. Yet in this chapter, this calm prelude, the story remained soft and contemplative. Creatures lived still and slow, guided by emerging sensations. The oceans hummed with subtle exchanges between cells and environments. Life was awakening, but gently, like the first whisper of dawn before the full sunlight of complexity. With this final piece in place, the beginnings of awareness, the foundations for all future evolution had been quietly prepared. When the Cambrian bloom arrived, it would draw upon every subtle innovation of these early ages. The structure of tissues, the coordination of organs, the emergence of ecosystems, the refinement of variation, the gentle shaping of natural selection, and now, the fragile beginnings of perception and behavior.